Yo. Yeah.
Good morning, everyone. I'm Dr. Jeanette Schnars. I'm the Executive Director of the Regional Science Consortium. I'm a basic background. The other side of the building is the research wing. Welcome to our 20th Annual Research Symposium. This is our exciting year for us. This is our 20th year. The RSC, just to give a little quick background, we're made up of 43 now member organizations. So that includes colleges and universities, state and federal agencies, school districts, and other nonprofits. And during a time when scientific research is more important than ever, we're excited to host our 2022. Over the course of the last couple of days now, we've had a great number of presentations, including 43 oral presentations, 58 poster presentations, and 20 art exhibits. So this is definitely a banner year for us to celebrate our 20th year. The presentations are given by undergraduate and graduate students, researchers, artists, and faculty, and so thank you to everyone. In addition to the in-person presentations, all the presentations are being live streamed on the RSC YouTube channel, so that's RegSciTV.com, and the presentations are being recorded. So if there's anything that you see in your proceedings that you missed that you'd like to see, the recordings will be up in a couple of days, and you'll be able to go back and see them. If you have any questions over the course of the day, today's the last day, please don't hesitate to visit the RSC staff at the registration table in the lobby, and they'll be happy to help you with anything you need. Please to start our final day of the symposium, let's see. And we're going to kick it off with a presentation by Tara Duffy, and she will be presenting on the effects of water chemistry on bosnian reproduction. Tara?
For the remote, the big button is the laser pointer, forward, backward, or you can use the buttons on there. Whatever's easiest. Perfect. Thank you so much. Great.
Alrighty, good morning, Regional Science Consortium Symposium attendees. My name is Tara Duffy, a freshwater and marine biology major and chem minor from Gannon University. Thank you for joining me today on this early morning. So following a year of concurrent research working with zooplankton, I am here to share my research on the effects of water chemistry on bosmina reproduction. This is the overview and research process that I'm going to take you through today, starting off with foundational knowledge to build into the methods and aims of my research, and finishing off with my major conclusions and the implications of my work. Presque Bay is at the forefront of this study. Belonging to Lake Erie, the shallowest and smallest of the Great Lakes by volume, it has the ability to have greater light penetration and change more quickly based off temperatures, and it's a very highly productive system. It is home to a diverse array of fish and bird species, supporting both recreational and economic profits. Bordering Ohio, Pennsylvania, New York, and my home, Ontario, Lake Erie is prone to receiving inputs from various anthropogenic influences as it acts as a sink receiving water from Lake Michigan, Huron, and Superior. The bay, Presque, a harbor snuggled right beside the city of Erie and closely near Gannon University, creates an ideal research environment for us at Gannon University and scientists. Zooplankton samples are one of these recorded measures that we take from Presque Bay with the environment. Particularly for my research, zooplankton samples were collected from the bay from May to October in 2023. Samples were collected using a 20-centimeter vertical mesh net with 153 micrometers lowered to the bottom of the bay, which is at a depth of about 5 meters, and then pulled to the surface. Zooplankton are described as the aquatic microorganisms that reside in water columns. In the Great Lakes, they uphold a vital position within the food chain as they serve to transport the energy from primary producers, like phytoplankton, which they consume, to secondary consumers, like larval fish, which eat them. Additionally, holding the role of a primary consumer makes them extremely sensitive to changes in water conditions. Disturbances that alter the composition of zooplankton communities in this type of ecosystem can be reflected in the overall health of the entire system. Zooplankton from each monthly sample were examined, analyzed, and classified under a microscope. I was able to identify and count various species of Bosmina, Leptodora, Eubosmina, cyclopods, and Daphnia, to name a few. A dominant species that I was able to observe in the bay were Bosmina. At the focal point of my research, I examined Bosmina females at various points of reproduction stages and identified eggs and brood pouches as asexual or sexual, then determined the number of eggs per individual. I wanted to see what seasonal changes would cause effects in Bosmina reproduction and trends. This line graph depicts the abundance of Bosmina that I recorded in the bay. These values were established by diluting the collected sample to 100 milliliters and examined a subsample of 10 milliliters. The trend we see here is data plotted reflected ideal conditions for Bosmina inhabitants in the months of May and October, reflected in the populations that's heightened on the graph above, and factors that inhibit their growth can be seen in June, July, and August, where the population's at its lowest. This graph puts into perspective the role and influence that Bosmina play in freshwater systems, particularly in the bay that we looked at today. We can see that in the handful of months that they are present in, like May, September, and October, their presence is significant, making up the majority of zooplankton species available in the bay at that time. That being said, understanding these distributions and abundance trends of Bosmina in the bay over the 2023 period will help us and me to better understand the Bosmina reproduction trends. In order to accurately identify both male and female within Bosmina within the sample, I needed to identify their main differences. So firstly, we can see that the males all appeared to be significantly smaller than the females. On a range, we can say 0.3 to 0.4 millimeters, whereas the females were closer to the 0.6 millimeter mark. Secondly, males have a notched first antennula, whereas the females have shorter antennulas and no notch, as well as a copulary hook right here that the male Bosmina will use for sexual reproduction. Lastly, and most prominently, males lack a brood pouch. So we have two documented Bosmina here from the bay, both from the October sample. And we can see that the male shows the first antennula, the hook, and lacks the brood pouch. This is a female Bosmina, and it just establishes that there's a visible difference that exists between the two sexes, regardless if there are eggs available in her brood pouch or not. So I identified and counted both male and female populations. This graph illustrates Bosmina as a female dominant species in Presque. In the months of June, July, and August, not one male Bosmina was recorded or counted. Bosmina are also opportunistic species, as they are capable of reproducing sexually and asexually depending on environmental stimuli and conditions. The female dominant populations are a result of parthenogenetic reproduction. This reflects that conditions were unfavorable for sexual reproduction, thus the females relied on the development of unfertilized eggs in the female brood pouch. The most males were recorded in the October samples, but only still accounting for just over 10% of the whole population. From this graph, we can assume where sexual and asexual reproduction is taking place based on the availability of males. So we can see that's more in October and September, the later months, and based off seasonal influences as well. Due to the differences in how both eggs originate, parthenogenetic eggs and resting eggs appear different in brood pouches of Bosmina. In the absence of mating, parthenogenetic eggs are capable of developing into offspring. This is ideal for species such as Bosmina, as it allows for means of rapid reproduction in favorable conditions. Additionally, we can think of, in relation to Presque Bay, The constant fluctuation of the environment, as it's the shallowest and smallest of the lakes, is constantly changing. So this ensures that they are able to reproduce in ideal and non-ideal conditions. So we can note the distinct circle shape, thin walls, and how they appear fairly small in that middle bosmina right there. These are examples of resting eggs present in the brood pouch of female bosmina, both observed in October samples as well. These types of eggs are sexually produced in unfavorable conditions with the goal of withstanding environmental disturbances or harsh conditions. They are capable of remaining dormant over long periods of time, common for overwinter conditions. Due to that, they present to have thicker and more rigid protective shells. Additionally, in the Presque Bay samples, the resting eggs appear to be larger and darker and appear to only have one resting egg in the brood pouch. In each Presque Bay sample, the number of present eggs were counted. These numbers reflect the raw data, or the number of eggs that were present in the subsample of 10 milliliters. Samples with eggs present in May, September, and October can show us that the water column conditions in the bay support reproductive trends of bosmina in these 3 months. The average number of eggs per individual in which eggs were present in was 1.17, with a standard deviation of 0.11. Reflecting extreme closeness of data here. Thus, individuals carrying 1 and 2 eggs were the most common. Literature data based on research from Lake Superior indicates that bosmina can carry up to 9 to 12 eggs. However, this data is based on bosmina residing in different geological lake communities with varying conditions. For instance, Lake Superior is the deepest and largest of the Great Lakes, thus composed of different water chemistry variables, supporting the idea that bosmina's health and reproductive trends reflect their environment. The observed eggs were categorized into one of 3 categories, whether they were parthenogenetic eggs, resting, or hatched eggs within the brood pouch. We can see that parthenogenetic eggs are the most abundant in all examined samples. We can see the presence of resting eggs only begin to occur in October as well, right here. These are examples of bosmina species with hatched eggs in their brood pouch, identifiable by the compound eyes in the brood pouch. And these are hatched from asexual means as well. Temperature also has a relationship with the number of eggs produced monthly. Oops, sorry, went ahead of myself there. So using YSI data, I took the temperature average from the water column and compared it to monthly total bosmina counts. The graph reflects great abundance of bosmina in cooler temperatures, which we can see in October and May. And they were the greatest abundance when temperatures were between 12 to 15 degrees Celsius. Temperature also has a relationship with the number of eggs produced monthly. Again, as temperature decreases the most in October, down to 12.9 degrees Celsius, we appear to see the highest rate of reproduction. This further establishes that bosmina favor cool temperatures in Presque for reproduction, and the graph highlights this inverse relationship. Again, pulling the YSI data for dissolved oxygen this time and graphing that against bosmina abundances, we can see a relationship as well. With greater DO measurements comes more bosmina present in the water column. DO data is commonly used to determine the water quality of a system and the health of a system. High DO concentrations reflect the presence of great amounts of life as photosynthesis is taking place and oxygen is available in the aquatic system for organisms to rely on. With colder water comes greater DO concentrations, which we can see the relationship on a seasonal basis here. We do, however, see a major inconsistency in the DO data in July. I hypothesize that human activity is behind this inconsistency and the drastic decrease in dissolved oxygen in the bay during the following month in August, right here. For example, Lake Erie is prone to eutrophication and harmful algae blooms. This could be the result of agricultural runoff or urban runoff making their way through the vast watershed and infiltrating the aquatic system. This may result in increased phosphorus or nitrogen concentrations in Presque Bay, which we know can contribute to eutrophication events. which would explain the oxygen change over this period of time as significant overgrowth would occur here, and then significant decomposition would occur here, removing oxygen from the water column, thus affecting the survival of Bosmina. Overall, this data supports that low oxygen concentrations can result in decreased abundances of Bosmina in the bottom waters of the bay and vice versa. With the assistance of dissolved oxygen measurements collected by the YSI data, we can also see that decreased availability of oxygen within the water column during the peak months of July, August, and September, with an emphasis on August, reflects maybe stratification is occurring here in this aquatic system. Thus, the research supports that decreased oxygen levels negatively affect the ability of Bosmina to survive and reproduce at a depth of 5 meters in Presque Bay. On the other hand, the high concentrations recorded in October support high abundances of Bosmina reproduction rates here. Also, for the eutrophication event that I mentioned, the overgrowth is a result of that. The prevention of light through the water column would be a result of a eutrophication event. This is reflected in this data depicting the sucky depth readings. August does not follow the parabola trend here, which we see, which could show a decreased light penetration reflected here, and thus supporting the eutrophication event that I hypothesize. Alongside this data, when examining samples under the microscope, I was able to see apparently high concentrations of algae under the microscope, which led me to believe that this event may have occurred. YSI data for DOC at depths of 5 meters was also taken into consideration within my research. However, this data proved inconclusive in my findings and reflects that DOC values do not greatly influence the survival or reproduction trends of Bosmina. The same goes for this graph, which depicts DOC data against the number of eggs counted in each sample. We can wonder what disturbance or event took place in September to make the DOC values decrease so rapidly into October. But again, no visible relationship here exists between reproduction trends and the dissolved organic carbon. Some literature experiments suggest that decreased concentrations of DOC play a direct role in increased phytoplankton biomass. So if that is true, we could reflect that increased food available for zooplankton species like Bosmina is causing its abundance to increase in October and making more ideal conditions for reproduction. PAR readings, or photosynthetic active radiation, refers to the amount of light that a system has for the use of photosynthesis, or usable light for organisms. I would hypothesize before completing this research that a direct relationship would exist between Bosmina population trends and PAR readings. Bosmina rely on primary producers, organisms that need sunlight for photosynthesis. I would think with greater PAR values would come greater food availability for Bosmina, relieving the pressure of food and allowing them to thrive. However, the opposite trend was observed here. We see from this graph that when PAR data is at its peak in July, Bosmina populations are at their lowest. Roles reverse when it comes to October, as the Bosmina abundance increases, but PAR reaches its lowest recorded values. This aspect of my research surprised me and is definitely something I want to look at in the future. The major conclusions and results established in the effects of water chemistry on Bosmina reproduction are as follows. Bosmina play a crucial role in the zooplankton biomass present in Presque Bay during May, September, and October months. Biomass are female-dominant species. Being opportunistic, they are able to reproduce both sexually and asexually depending on environmental conditions. This is reflected by the greatest number of observed eggs being parthenogenetic. An inverse relationship exists between Bosmina abundance and temperature. Populations of Bosmina were greater when temperature was between 12 to 15 degrees Celsius, reflecting their preference of cooler environmental conditions. The same relationship exists when it comes to reproduction. More eggs are observed in the coldest of the studied months. A direct relationship exists between DO data and both Bosmina abundance and eggs. Relying on the availability of oxygen for both growth and reproduction are Bosmina. I hypothesize that disturbances like eutrophication events in water systems negatively affect the ability of Bosmina to survive and reproduce due to the alterations of available nutrients, decreased oxygen, and decreased sunlight penetration through the water column. This research process proved to solve an array of my initial inquiries that I strive to understand better. For instance, the role of temperature, dissolved oxygen, PAR and light data on the abundance of Bosmina in Presque Bay, and how these water quality variables influence Bosmina reproduction trends and rates. However, this process also uncovered a number of additional questions that have sparked my interest. In the future, I hope to take this research to look at particular nutrient data like ammonium, phosphate, and nitrogen concentrations. I was surprised to learn that the light data via the PAR readings had little influence on Bosmina survival. That being said, I strive to further understand other factors that may influence food availability or contribute to the favorable conditions in which the zooplankton species can grow and reproduce in. In addition to better understanding the anthropogenic influences and how dominant zooplankton species like Bosmina may be impacted in the future by continuous human alterations and effects on aquatic ecosystems, as well as exploring the more extensive reproductions that this may have on food chains and larger, and how those would be altered. But most importantly and most interestingly, as many biology faculty at Gannon University start to explore Lake Pleasant, water chemistry and biological makeup, I think it would be extremely fascinating to analyze zooplankton samples retrieved from this lake and complete the same examination of its Bosmina populations, being able to compare and contrast the trends observed in Presque Bay to another lake located in Erie with drastically different geographical factors and anthropogenic influences could prove to share a lot more about the Bosmina species and how their reproduction rates respond to water parameters. Thank you all for your time today. I hope you enjoyed my research on the effects of water chemistry of Bosmina reproduction as much as I did. Thank you so much for listening.
I was just curious, you had a figure where there was the abundance of other zooplankton was really high. I think it was in August A and B, but the Bosmina was like 0. I'm just curious what species, if you know, were present during that time.
Yeah, interestingly enough, it was Bosmina and Daphnia, which I saw the most in that month. I think it was August, like you said, which was really interesting. And I know I was talking with my professor about it. It would also be interesting to do the same study on another species and figure out what water quality parameters they need in order to reproduce or thrive in their aquatic environment. It's interesting how it's different for zooplankton, but.
Well, kind of connected to that question, I was going to ask, do you think the living, like the contagious stuff, is the same for the other type of species that you mentioned, like the Daphnia?
Yeah, that's a great question. Honestly, I think that's like a next step, looking at like how long they're surviving in the water column, what's affecting their length of survival. It's definitely something I could look into, but.
Thank you very much.
Thank you.
our next presentation is by Dr. Bumney Aloy, and we will be hearing about the effects of, I can't read this from here.
Don't worry about it.
And co-herbicides on aquatic organisms.
Hello guys, good morning. Like you said, my name is Bumi and I'm here to talk to you about safeness. Anyone heard about safeness before? Pretty strange thing, yeah. That's how chemicals that are not well known are quite strange before they become a problem in the aquatic environment. So my focus is on aquatic environment, and today I'll be talking about evaluating the effect of safeness and the co-herbicide on aquatic organisms. So I'm trying to look at the toxicity assessment and the recovery insight. It is important that when organisms are exposed to toxicities, we should be able to measure if they would be able to recover after facing all of those things in the environment. So I'll start by telling you what safeness are. All right, so we know that in the quest to ensure food security, we use herbicide to clear the weeds so that our crops are not affected. But over time, research showed us that herbicides are able to cause injurious effects on our crops. So what did scientists do? They formulated safeness from the word safe. Safeness are formulated with herbicides to protect the crops from the injurious effects of the herbicides. Got that? So safeness will be formulated together. When you spray them on a farmland, they'll protect your crops from the effect of that herbicide. But they will not take away the effect of the herbicide in the ability to clear the weeds, but they are just protecting the crop so that we have maximum yield and everything goes well. Well, we were told that safeness are inert. That means they're not active. We are not expecting them to be toxic in the environment. As a result of that, it's been used, anyhow it's going to be used. Farmers are just using them because we want maximum yield. From the picture you can see there, I don't think it, I am sorry. Maybe I will not use that. Yeah, from that picture, we'll see when we do not apply safener or herbicide, crops will literally go well. But unfortunately, the environment does not encourage, a lot of factors will affect whatever you plant in the environment. And when you use safeners alone, the crops are doing well. Herbicide alone, you can see we do not have much yield because the herbicide is injurious to our crops. And when we had safeners plus herbicide, we have our maximum growth again. Okay, but why, excuse me, why are we bothered? Accumulated safeners and herbicide has been found in the aquatic environment. Just like the case of microplastic and PFAS and all of these things going on around. We see them in the environment. But because we do not know so much about them, we are thinking maybe because it's inert, it's not toxic, we should not be worried. But at a point, scientists will be worried. Everything that goes into the water body, we want to know if it's affecting anything, biodiversity or all of these things. So we believe it can pose threats to our aquatic organisms. And that's why we are going to be doing this study. So I am thinking my students will be able to assess this in the course of the coming years. So maybe next year, whenever our conference is, we can present the results to you guys. So the presence of these chemicals in the environment is the major reason why we are going to be looking at the study on non-target species, because they are not targeted towards daphnes or zebrafish or any of these species in the aquatic environment, even zooplankton and phytoplankton. But then we are interested in seeing them. We all know how all of these chemicals could go into the water body, either by leaching, dissipation, water view, water runoff, everything. That's how they get into the environment. Where they could cause environmental pollution, contamination, or even the death of organisms in the environment. All right, part of the justification, we do not have so much things documented about safeness effect on organism at all. We do not have. Now, recently, I think 2019, CV and his colleagues have been looking into safeness, so they have been doing some research. But most results you would see would be on plants because they apply to plants. But whatever applies to plants might apply to all of the other organisms in the environment. So my thought would be looking at this on algae, on zebrafish embryos, because I know we are not, there are a lot of ethics thing around using zebrafish as a vertebrate in research. So we'll be using Daphne also as an invertebrate, and its study we assess the toxic effects of safeners individually and in combination. Every safener has a herbicide formulated for it. So they have, if for example, safeners like methylphenidate, don't worry, those are big words. It has its own, herbicide that is formulated for it. One of those herbicides is phenosapropyet. Don't worry about it too. They are big names, so I'll call them Fen in the course of this presentation. All right, my proposed animal model would be daphnids. You know all about daphnids, water fleas, and zebrafish embryos. So why did I choose those? These two organisms have their whole genome sequenced. So there's literally nothing that, will happen with them that you would have not known. You will have something to fall back on in literature. And that is why I'm choosing them. You can see they're transparent too. When you put them under the microscope, you can literally see what is going on with them. I have used them. If daphnids would not reproduce, you will see all of the eggs in there and would never come out. It will not, lay those eggs. Zebrafish embryos, I've used them also. You see deformations. You see oedema, and they would not, come out of the egg yolk. So that is the major reason I'm using them. I have a lot of literature about them. I can see what is going on with them, and they are very easy to, to raise, to culture. We don't have much stress culturing all of those. All right, how do we want to go about this study? Now we are moving towards eradicating the use of live animals, right? So that's why I'm using zebrafish. But first, we'll do some in silico prediction. These predictions, would help us to use some scientific or computer software to do simulations instead of using biological studies. So I have used this before. We have one that is auto-dock. When you dock safeners and, whatever endpoint you're looking at in all of those organisms, say for example, ashin in zebrafish embryo, there is this enzyme called ZHE1. You dock them together. If the binding energy is great, you would know that it's possibility that this chemical can affect ashin in zebrafish. So I would first test all of those in silico. If I see any binding affinity that is greater than what I would think, then I'll go ahead in the laboratory knowing that I will see a particular result. So we want to use this to narrow down what we would see. All right. We're going to be using the selected safeners plus the co-herbicide to predict their effect on the endpoint, and it saves time and effort. It's less expensive compared to biological studies, even though it has not been accepted yet to use just in silico prediction without biological studies, but then we will try this first and see. How do we go about it? We're going to be doing acute and subchronic studies. We are not going to expose them for a long time, because we are careful of, what it's going to do to them and euthanizing them humanely and all of these ethics surrounding biological studies. And the endpoints we'll look at include survival, mortality. We look at reproduction, especially in daphnids. We will not be able to see the reproduction in zebrafish. It takes a while compared to daphne. On the 8th day, daphnids should be reproducing. So we would be able to see. So in a matter of 3 months, you can do about 5 generations of daphnids. So that helps us to get a lot of information from them. We're going to look at behavior. When daphnids are exposed to some contaminants, you see them, the swimming pattern changes. They would be in the same space, swimming in that space, up and down that way. Something is wrong with them. You cannot see that because of their transparency too, just like I mentioned. Biochemical activities, we're going to do that to measure the stress and the defense responses of these organisms to this chemical. So the use of catalase, the use of SOD, superoxide dismutase, GST, all of those things we would be able to use. And it didn't stop there. We're going to do a recovery studies on them. After exposing them for a while, we bring them out from the chemical we exposed them to, put them in the fresh water, allow them to eject some of the chemicals if they would be able to do so. So we will now measure all the endpoints again and see how much they are able to eject these chemicals from them and see if they have the capability to even do that at all. So while we will test reproduction on these ones that we are trying to recover, we will see if their reproduction gain will be the same like before they were exposed, while they were exposed, and after they are exposed. We assess variability across species that we will use. I'm thinking of RJ2, using all this fluorescence and all of that, counting the RJ and seeing what we'll see. Because RJ, primary producer, they are usually very direct, coming in contact with this contaminant faster than other organisms that will be at the depth of the water. All right, that is all we are going to be doing. So thank you for listening, and I hope the result will come. I will be able to share with you. I have a paper on safeness if you want to look at it. That's the QR code you can use to access it and just see before and get familiar with safeness. Safeness might be the new PFAS coming. 25, 26, 27 years ago, my supervisor, the first to discover PFAS, he mentioned PFAS, but it was taken to court. There were a lot of problems. Oh, you're saying something, but now it's a problem, and everybody is looking into PFAS. I'm not saying safeness will be like that. They are designed not to be active, but I have done a study, and I discovered they were active. They were active and toxic. Even safeness were more toxic than the herbicide itself. But well, at the end of the day, mixing them together minimize the effect on the organism. So we're going to continue this study, and hopefully when we see next, I'll bring the result to you. Thank you for listening.
Questions?
Yes, please.
What are safeness generally made of?
Just like the herbicide, they're chemicals.
I didn't know if there was like a specific one, or if there was just like such a variety of them.
Oh, there's such a variety of safeness. So the earliest one that were used, like dichlomide, like dichloroacetamide, they belong to different classes. Okay. So they're not just one and all, different classes, because just like we have chemicals having CH, all of that, and all other ones having OH attached to them in their structure, that's the way safeness are too. Yeah. Yes, please.
Do you have some safer herbicide combos that you're going to focus on with your initial study?
Say that again, please.
Are there specific safers or herbicides that you're going to
And that is what I...
prioritize?
So I have been looking at mefenpine and cyprosulfonamide. These two safeners are used on grains. In Pennsylvania here, we grow corn. Cyprosulfonamide is used on corn almost all year round. So I'm thinking if we could go to water bodies very near the farmlands where our farmers are planting this stuff, we will be able to measure the concentration of the safeness in that water. Then we will use that relevant concentration in the lab, and we'll see what will come up with what we have. Yeah, so I have that. But the common ones too, my mind is going there, like the dichlomide is being used a lot in the past. The one I mentioned, the first two one are emerging safeness. So we are just hearing about them. So farmers are using a lot of them. So yeah, I'll be looking at those. Yes, please.
What was the computer simulator that you used to like get this information from?
Okay, so molecular docking, you could use AutoDock. I have other tests like the ADMET test, like the toxicity estimation software test, TEST, it's a lot. Most of them are free online that you could access, and the ones that you have to pay for. But if you students and all of that, if you don't have the money, maybe your professor can be the one to buy using the money from the school, of course. But yeah, AutoDock for molecular docking. On top of my head right now, I would not remember all of the other docking, whatever. But if you Google them, yeah, you will just find them.
Sorry, I have a follow-up question.
Oh yeah.
Do you think when you, like maybe later in the future, you would do it like actual using like the biochemistry and stuff like that instead of the simulator, or would you just continue keeping the data?
Not science is it's not dogmatic. We could use several things. We want to see results. We want to see what it will cost. So yeah, it just shouldn't be about the molecular docking alone. The docking is just to see what's the binding energy. Are they docked together? The ligand and the substrate, are they coming together? If they're coming together, I would think, okay, maybe they would bind whenever I'm doing that experiment. It's just to, it doesn't give me a definite answer of how much toxicity is going to happen. Is it going to cause deformation and all of that? It's just a prediction. And science does, we predict, but we have to test what we predict to get the answer, yeah. Be the first one.
Thank you so much.
Yeah, thank you for listening.
All right, our next talk is Amped Up, and Elise Rinke is going to present on the microbiome on bullfrogs.
All right. Hello, everyone. My name is Elise Rinke. I'm a graduating senior from the University of Pittsburgh. And this past summer, I had an awesome opportunity to complete a research fellowship down in Pymatuning State Park at the University of Pittsburgh's Pymatuning Laboratory of Ecology. So in this project, I worked with a graduate student studying the impact of antimicrobial peptides and the skin microbiome on bullfrog thermoregulation and also chytrid fungus disease resilience. So this project was created in collaboration with Riveter, the Resilience Institute Bridging Biological Training and Research, which also has a fantastic acronym, I have to say. This is an institute dedicated to researching and combating the causes of global amphibian declines. Riveter has multiple sites across the Americas, which you can see here, and then obviously the site that I was at this summer is the Pennsylvania site down in Pymatuning. The Pennsylvania site poses a very unique opportunity to conduct research because most of our native amphibians are very resilient to chytrid fungus. So we're not seeing these massive die-offs that chytrid fungus is causing in pretty much all of the other Riveter sites. So we might ask, why is it so important to have an institute dedicated to studying amphibian resilience? This is because amphibians are facing significant threats that compound to cause global declines. One of these is climate change. Amphibians are ectothermic, meaning they get all of their heat from their external environment. Thermal physiology has many established links to resistance to pathogens and other environmental stressors. So as climate patterns change, we could also see the thermal physiology of these amphibians changing. And research really isn't adequate yet to show what the impacts might be to amphibian populations. They're also suffering from habitat loss and fragmentation. A lot of amphibian viable habitat is being broken apart, lowering gene flow and potentially even causing genetic bottlenecking, which is an additional stressor that these amphibian populations have to face. Next, invasive species can often outcompete the native species for resources. And there are many emerging diseases, many of them spread by human interactions, particularly the wildlife trade. These include ranavirus, B-Sal, and Batrachochytrium dendrobatitis. These last two diseases are both known as chytrid fungus, but the one that we'll be focusing on today is Batrachochytrium dendrobatitis, also known as BD. So we'll talk a bit more about the BD life cycle. BD is a skin pathogen that has absolutely decimated global amphibian populations. It enters the skin where it grows and reproduces, causing symptoms ranging from ulcers, convulsions, weight loss, and eventually even death, as you can see in the pictures over here. As it grows and reproduces, it eventually releases spores, which are actually able to swim through the water to encounter a new host. And it's a very generalist pathogen, so it can travel from amphibian species to species. It also is able to survive in the soil for a very long time, months to even years, and the true extent of how long it can survive in the soil is still somewhat unknown if environmental conditions are ideal. So it's pretty scary when we're thinking about some of the species that have gone extinct in the wild and are currently being bred in captivity with the hopes of eventually reintroducing them. But if the pathogen can remain in the soil, even if there are no hosts there for it for years, it kind of makes us lose a little bit of hope as to when we'll be able to reintroduce these species. But as I said, a lot of research still needs to be done, so we're really not sure the exact dynamics of this. Then these pictures here were all taken in the Sierra Nevadas. If you remember from my previous slide, there's also a Riveter site there studying chytrid fungus. The one on the bottom is one of my favorites. It was taken by an awesome photographer, Joel Sartori, who I emailed and asked if I could use it in my presentation because I just thought it was so cool. but I would definitely recommend checking out some of his photographs. They're very powerful wildlife photographs. So a lot of areas in the United States are really not suffering from chytrid fungus, like certain regions, but obviously the Sierra Nevadas here are still seeing those declines. And population declines from chytridiomycosis have been described as the most spectacular loss of vertebrate biodiversity in recorded history. This figure from Shiel et al. 2019 shows impacts by global region. The paper concluded that 501 amphibian species are experiencing declines, and 90 species have likely gone extinct because of chytrid fungus. I want to note that there are just different levels of biodiversity in each region, so that does contribute a little bit to these varying levels that you see in this figure. However, even if we account for this, South America and Mesoamerica are still experiencing the most significant declines proportional to the species that they have. This relates to the intermediate virulence hypothesis, which suggests that over time, a host species will evolve to be less susceptible to death from a disease, while at the same time, a pathogen will also evolve to be less deadly, or else they would risk killing off their entire host population. In Central and South America, chytrid fungus was introduced fairly recently, so there hasn't been time for that coevolution to occur. But in many regions in North America, chytridiomycosis seems to be much less severe. Now, as we saw earlier, this isn't universally true. Declines in the southwest around Sierra Nevada ribbiter sites are still pretty severe, but overall, chytrid fungus seems to be much less deadly in North America, with pretty much every species being resistant, including the focal species of our study, the American bullfrog. This makes it a really great study system because we can analyze dynamics of the disease in hosts without worrying about mass fatalities. We can look at relationships between chytrid fungus load and parts of the immune system over a longer term than other sites might allow. And the parts of the immune system that we studied this summer are antimicrobial peptides known as AMPs and the skin microbiome. AMPs are produced in the granular glands, so you can see those right here, and they're secreted onto the skin to fight against pathogens that may encounter the skin, which, if we remember, chytrid fungus is a skin pathogen, so that would be one example. One of the most common times this response happens is after a predation attempt. The dermis contains mucosal glands that produce mucus and make the skin really slimy. So if you've ever gone frog catching and you catch a frog and it gets really, really slimy, that's actually because it thinks you're a predator and it's trying to escape from you. At the same time, though, AMPs are also released from these glands in case an injury were to occur in that predation attempt. That might cause a cut from clawing or biting that would make the amphibian more susceptible to infection. So the AMPs are released kind of proactively to prevent any infections that might happen. We can also see in this figure, in the green up here, commensal bacteria that live on the skin. So these constitute the skin microbiome. And for a very long time, it was kind of believed that the skin microbiome was pretty neutral. It didn't really help anything, but it didn't hurt anything either. More recent research has shown that the skin microbiome might actually be one of the first lines of defense against skin pathogens, and it can have really profound implications for the innate immune system. They've found that a lot of the microbes that live on skin produce metabolites that can actually directly fight disease, and it's been hypothesized that some of these secondary metabolites also upregulate AMPs, so they're actually increasing other parts of the immune system defense. And then this image I just really liked because it shows an actual photo of the granular gland right here, the epidermis up here, and then AMPs would be secreted from here onto the skin, as well as mucus. So now that we have that background, we can kind of loop back to the more direct focus, thermoregulation. As I mentioned, frogs are ectothermic. So their thermoregulation is completely behavioral, with basking being the primary means of obtaining heat. We know that frogs that thermoregulate at higher temperatures have more active immune systems and are more resilient to diseases. If their thermoregulation tends to be higher, they'll produce more AMPs, they'll have more active skin microbiomes, and other parts of the immune system will just upregulate to lower disease loads. Our research, though, aims to uncover whether this is a positive feedback loop, with AMPs and the microbiome also upregulating behavioral thermoregulation. There are a lot of unknowns about how AMPs and the skin microbiome function, and just generally how parts of the frog immune system function as a whole. So Ribbiter has for a long time proposed that this could be one of the pathways that these parts of the immune system actually prevent disease. So this leads me to our research question. How do AMPs and the skin microbiome impact thermoregulation and chytrid fungus resilience in the field? I'm going to walk us through the methods, and then we'll get into some of the preliminary results. I will warn you that a lot of this data is still being analyzed, so we don't have a full set of results yet, but I'll also talk a bit about future directions and the things that we are still working on. As I mentioned, our research was conducted in Tuttle Pond, right down here in Pymatuning. If you're not sure where that is, it's kind of an inlet off the Pymatuning Reservoir, just about 45 minutes south of here in Pymatuning State Park. So we collected 60 American bullfrogs from Tuttle Pond to assign to treatment groups, housing them in enclosures like these in the labs for the brief period that they were being treated. To walk us through the treatment groups, I put this diagram in the middle here. You can see AMPs in red, and then the commensal bacteria in yellow. The first group is a control group that received only placebo treatments and maintained a fully intact antimicrobial peptide and skin microbiome community. The second group underwent depletion of the skin microbiome. So this was done by soaking the frogs in an antibiotic cocktail for 48 hours, and then at the end we swabbed the frogs to test their microbiome just to ensure that it was depleted like we thought that it was. The third group underwent AMP depletion, which was done by injecting norepinephrine in the dorsal lymph sac, and this basically stimulates a biochemical pathway that makes the frog think it's being predated. So during this pathway, it thinks that there's a predation attempt occurring, and it basically just secretes all of its AMPs, which we were then able to send to a lab to analyze, and also the AMPs are fully depleted for about 45 days, which gave us plenty of time to go through the rest of the field season without them having that innate immune system intact. And then the final group has depleted AMPs and skin microbiome. The reason we might want to look at both of these together instead of just looking at each independently is because of the proposed interactions between AMPs and the microbiome. So as I mentioned before, there's hypotheses that the secondary metabolites produced by the skin microbiome can actually upregulate AMPs, so looking at both of them in conjunction allows us to kind of analyze these interactions. Then, once the frogs were fully treated, we released them back into Tuttle Pond, and each frog had a unique identifier, so we could track changes in AMPs, skin microbiome, chytrid fungus load, and temperature for each individual throughout the field season. This was probably the most intense part of the field work to me, because we constructed, you can see here, these huge enclosures. They were 4 by 6 meters, and we designed them ourselves throughout the summer, and we made 4 of them and had to deploy them into the ponds. So it was quite a task. We ran into a lot of problems along the way, but it all worked out in the end, and the frogs did not escape. Also, the reason that we had enclosures when we want to monitor these frogs in their natural habitat is because the pond was really big. So we were like, if we just release these frogs, they're gonna run away. We're never gonna find them again. So that was kind of the justification for why, even though we want to monitor them in their natural environment, we still have to have these enclosures to keep them contained. And then also, I'll note on the enclosures, we included 4 meters of a water portion to allow natural water behavior. They are water frogs, obviously, with some logs in there for basking, as well as 2 meters of a land portion that was vegetated to simulate that natural behavior. Then, to monitor temperature, some of the frogs were given temperature sensing belts, which were very cute. There's a small temperature sensor in each of these, and then this portion is a 3D printed case that keeps the temperature sensor against the frog's body 24/7, and then they're attached around the waist with a string. This was one of the most exciting parts of the experiment to me because this technique hasn't really been used to monitor temperatures of frogs in the field before. Mostly, temperature sensing is done with handguns, so you actually go out and manually scan the temperature of the frog, and it's kind of a hassle and you don't get nearly as many data points. But the potential problem, and the reason that we didn't give every single frog a belt, is because in frogs, thermoregulation is 100% behavioral. And having something around their waist that they don't normally have could very feasibly alter frog behavior. So by giving some of them belts and some of them just monitoring by hand, we're hoping to be able to analyze these differences and see if there really is a behavioral change that's caused by the belts. And I'll also note that we did calculate the weight strategically so that it wouldn't be a hindrance to the frog in any way. This picture also highlights another unexpected problem that we came up with as we were deploying these enclosures, and it's that bullfrogs can climb, which I never would have expected. It was something that none of us anticipated, But at 2 a.m. when we went to release the frogs into their enclosures, they all started trying to climb out. So we spent like 3 hours in the middle of the night adding lips to the enclosures and making sure that our frogs didn't escape, which they did not. So then finally, I'll go through the data that we collected while these frogs were in their enclosures. Every day, we monitored temperature of the unbelted frogs with handguns. We also monitored mass to track if the frogs had any health problems, and finally monitored body length, mostly just to confirm the identification of the frogs. Every week, we did a full capture survey where we took swabs to test for chytrid fungus load and microbiome composition. This brings up another very interesting future direction, because not many studies have tracked microbiome regeneration with fungal load in the field in a non-sterile environment. So as we get that microbiome data back, we're really interested in looking into it and seeing if there are certain microbes that recolonize faster, certain microbes that we can more strongly link to chytrid fungus declines, and all of that information. So data analysis for this project is very much still in progress. However, I was able to run some very preliminary analysis on the relationship between immune function depletion and thermoregulation. On the y-axis over here, you'll see the average temperature for each of the groups listed on the bottom over the course of our monitoring period. And then you can see here, the temperatures between different treatment groups seem to be pretty similar. I will note that we haven't been able to go in and account for confounding variables like mass, body size, etc. So some more clear patterns might emerge as we start to account for these variables. However, as of now, our results indicate that there's really no relationship between AMP and skin microbiome and thermoregulation. So even though we found no relationship in this preliminary analysis, these results still give us valuable insight into how the immune system functions. Scientists at Ribeter have a long time proposed that AMPs and the microbiome might impact behavioral thermoregulation. So our results, once they're fully analyzed, might be able to rule that out as a potential pathway. Once this data is compiled, we're also hoping to track regeneration of specific microbe populations with chytrid fungus and see if we can identify any other significant patterns that way. Then another big data point that I mentioned before that we're still looking into analyzing is thermoregulation differences between belted and unbelted individuals. The belts gave us big data sets like this that show temperature every 15 minutes for the entire time that the frogs were wearing them. This is just a 3-day period out of the whole 3-week monitoring period that we had. So this is a really valuable insight because, as I mentioned, people haven't really used this temperature sensing belt in frogs before. So if we can pull data points from here that were taken at the same time that we did manual sensing in the field and analyze if there's a difference in temperature, we might be able to show that these belts don't really cause, a significant behavioral change. And that could make it a really promising tactic going into the future. So these are my sources. As I said, there's still a lot of data left to analyze, and we're very excited to see the rest of the results. And I want to put out a big thank you to everyone who helped with this project, including Lucy Anaya, who was my advisor over the summer. If we have any questions, I'm not sure if we have time to take them now. Yeah.
Is chytrid fungus more severe for frogs of different sizes or age, or is it kind of not discriminating?
Yeah, I'm not really sure. I know that a lot of the amphibians around South America tend to be on the smaller side than the ones we have here, and South America is the area that we're experiencing the most significant declines. I think the more prominent factor is how recently the disease was introduced. So just overall, in areas that it was introduced more recently, pretty much all of those frog species are experiencing more severe declines than in places that it's been around for longer. Any other questions? Yeah.
Was there a limitation on which frogs you could put those temperature sensors on? Say like a size?
There was, yeah. We had to weigh each of the frogs and make sure that they were an adequate size, that it wouldn't like hinder their behavior too much. So that's part of the reason we haven't been able to analyze it yet. We want to make sure we're comparing frogs of similar sizes when we do that analysis. Yeah. No, there were not. There was a minimum size that the literature had kind of showed would be a size that wouldn't hinder the frog's behavior. So they were all the same size belt, but we just made sure that the frogs were above a certain mass. Yeah, go ahead.
So, that fungus, is it just generally invasive and that's why it's harmful? Or is it just something that's harmful in the environment, it's everywhere and frogs have to deal with it?
Yeah, interestingly, it was mostly introduced to new places by the wildlife trade, specifically African clawed frogs that are from Africa, where we're not sure if that was where it originally evolved yet, but that's one of the hypotheses, and we know that it's been there for a very long time. So the amphibians there aren't harmed by it, but then when people get these frogs and release them into the wild, that disease is introduced to new populations. So it is an invasive thing, and the populations where it's more native to, where the frogs have evolved, we're not seeing these massive declines from it. Yeah, go ahead.
First of all, kudos for being out at 3 a.m.
Yeah, I am not a night person, so it was a huge adjustment for me.
Congratulations. That bodes well for graduate school. Do you think being in a small enclosure like that, is there a more stressful situation, or would that influence how much they secrete?
Yeah, that was another thing that we definitely considered, and we read a bunch of papers, a lot of them from the 80s when they were doing a lot more of these like behavioral-oriented trials that described the natural territory of each bullfrog, and it varies by sex. So we accounted for like the amount of females to males that we had and the natural territory that they would have if they weren't in an enclosure, and made sure that those enclosures were constructed at a minimum within that range.
And that leads to the next question. Males and female distribution. Did you say that in terms of the enclosures? How many have males or females? Or did it?
I did not. It was actually just what we ended up catching, but it was about 4 females to 1 male, interestingly. So a lot more females.
And how much do these belts cost?
I have no idea, actually. The graduate student that I worked with 3D printed the cases herself, so it was just the cost of the sensor, which I assumed wouldn't be too much, but I don't know the exact number.
Thanks.
All right, any other? Yeah.
Mine's slightly silly. Do they all have different colored belts?
They did not. They did not. I almost put a picture in here. We edited one of the belts to be like a Gucci belt, and I thought it was really funny. But they were all the same.
That seems like a great way to ID different frogs. They all have, different versions of their belts.
Yeah.
Any other questions? Very well, thank you so much.
All right. Thank you all.
Oh, yeah. There it is, right there. No, you're good.
Our next presentation is by Kira Armstrong and Katrina Orange from Gannon University, and they're going to give us an update on the bat population on campus.
All right, everyone, good morning. My name is Kira Armstrong.
I'm Chelsea Total.
Today, we're talking about a census of the bat community on the campus of Gannon University in Erie, Pennsylvania. Now, let's talk bats. So, bats are the only mammals that are capable of true flight. What makes them mammals is they have a mammary gland that produces milk for their babies, which is also known as pups, and they're actually giving live birth. Some people don't know that, but that's what makes them a mammal too. So they make up about 1/4 of the mammal species with 1,100 different species of bats. Now, that's a lot of bats, if I say so myself. They're considered to be nocturnal because they feed at night and they sleep during the day. Even though they pollinate flowers, disperse seeds, and eat millions of insects, they have been scientifically neglected until recently. But bat numbers are increasing because of a new disease affecting hibernating bats known as white nose syndrome.
White nose syndrome is caused by the psychophilic or cold-loving fungus known as Pseudogynoecus destructans. Over half of all U.S. bats hibernate in caves, and caves provide a damp, cool environment that allows for the fungus to grow. And this fungus usually manifests on the bat's tail, wings, face, and ears. And the fungal spores are viable for an extended period of time, so the bats will hibernate and then sometimes they'll wake up and the fungal spores will still be on their extremities. And here we have some images of the white nose syndrome fungal spores. On the upper picture, you have fungal spores on the bat's face, fungal spores on the bat's wings, and all over the bat's ears.
So WNS causes bats to wake up during hibernation using up their precious fat reserves. This can also suppress their immune system. So WNS doesn't actually kill the bat itself. This fungus is causing them to scratch themselves awake because, you know, if you're scratching yourself very viciously, you're going to wake up in the middle of sleep. So this is how they're waking up, but because they're hibernating and it's colder weather, they need to go replenish those fat reserves, right? So they're going to go leave the cave and try to find insects. But they're hibernating, so that means the weather is really kind of cold and not really the greatest. So all the insects are actually gone, so they can't replenish their fat reserves. So this will either cause them to starve to death and die, or because it's so cold, they'll die from hypothermia. Mortality has approached 100% in some cases. Let me say it again, 100%. So it's cold-loving fungus, and it loves the really cold and damp conditions that cave brings. So bats also love caves, so it's just it's a perfect way that the fungus can attach themselves to the bats so they can survive. So since 2006, WNS has already killed over 7.5 million bats. So 2006 seems like a very long time in human years, but for scientific research, this is very recent and very new. This is a big problem because in this quote down here, scientists are saying this is the most precipitous wildlife decline in the past century in North America, especially because bats really help us in a trifecta impact that Chelsea will go over later in the slide. So, let's see if I can get more split here. Okay, awesome. So over here in New York in 2006, this is where we first saw WNS. And you can see it started on the eastern coast of the United States, and it started to go into the bottom part of Canada on the eastern side. Then in 2016, it hopped all the way over to the western coast of the United States. And then over the years, it just gradually kept increasing and increasing, and it just started covering the United States and the lower parts of Canada. And this goes from 2006 all the way to 2023, 2024.
There's a concern of the spread of white nose syndrome as 9 species have been affected, 4 of which are federally endangered. So there's that threat of extinction in those 4 species. There's a trifecta of environmental, agricultural, and financial effects of the spread of WNS. And you can see in our little graphic, the more bats there are, the less insects. And the less insects means there's a lower need for pesticides. And if there's a higher need for pesticides, then there's more money that needs to be going in to fund the pesticides. But if we have less bats and more insects, then we need more pesticides and more money to fund the supply of pesticides. In our current strategy, our management and conservation of bats and their habitats that are in the path of WNS. Researchers at the University of Georgia have developed an antifungal treatment using the bacterium Rhodococcus rhodocrois, and it's an antifungal treatment that treats the fungus for white nose syndrome. And our rhodocrois has been shown to slow mold growth on ripening fruits. In 2015, 75 bats were treated by hand with this bacterium and released back into the wild, and were actually shown to have survived because of the treatment. And researchers are continuing to work in the Black Diamond Tunnel, which is an area that is affected by white nose syndrome.
So further investigations, Bat Conservation International is collaborating with researchers from Western Michigan and Ball State universities to test a newly developed antifungal agent, chitosan. So white nose syndrome is full of fungus, so we're trying to find an antifungal agent to kind of counter-react with it, so that way we can just try to find a treatment for it. And chitosan is one of those antifungal agents. We also have antifungal volatile organic compounds known as VOCs that are being tested, essential oils, and other compounds that are also being investigated as an antifungal agent. So development and testing is underway for a device to modulate all of these treatments. So like Chelsea said, they're hand treating the bats in the Black Diamond Tunnel. So they're trying to find a dispersal device like a sprayer to just spray all these bats, because if you spook one bat, the whole cave's going to freak out, and then you have no bats to be treating. So that's why they're trying to find a spray to treat several bats at a time and hopefully cover the whole cave. So why downtown Erie, PA? First off, it's Erie, PA. We all live here, so. Not only that, but right here is Long Point, Canada, and over here we have Presque. So this is the shortest distance from Canada to the United States, and this is in the path of the Eastern Flyway, where bats and birds migrate. So yes, some bats do migrate back down south during the colder months, and then some of them actually hibernate and stay in caves. So that's why it's really important to study at Erie, because we are along that flyway, and then it's also really important because we are from Gannon University, and we're in Erie, PA.
So a group of Gannon University students on this research project, there was a student researcher for each day of the week, and each day of the week at sunrise, a Gannon University researcher would go out on this designated bat walk that we came up with. That's surrounding Gannon's campus, and the walk takes about an hour to do, and we picked it mostly because that's where we anticipated to find bats around the campus.
So, marking bats. This is my favorite part of the research because I get to mark the bats, so I really love this part. So all bats were recorded by the building, location, north, south, east, and west, and what species of bats they are. So we mark bats with a little dot scheme of washable, non-toxic paint. So you'll see pictures of me actually marking the bat. But once we see a bat, we'll get a ladder because they actually roost in higher spots to avoid predators. And we're not allowed to handle them, so we have to get a paintbrush, put the non-toxic paint onto the paintbrush, and ever so gently just brush the bat with the paint. Not too much paint, so a predator will see it, but just enough that we'll see it in a higher spot. So this is done to see if bats usually roost in the same nesting area, because in 2010 there was a lot more bats on campus. Now you'll see the results that we only got 8 bats this year, so it's not really a big deal like the color scheming, but you'll see something really cool in the next slide. So Game Commission has strict guidelines on handling the bats for many reasons. I don't want rabies. No one wants rabies. So that's why we don't actually handle them. And then we don't want to disturb the bat. We don't even want to, hurt the bat. And then it's part of our research to not spread WNS. So it'll go against everything we're talking about today. Okay, so as you can see, these 2 bats were together roosting, so we definitely want to mark them with different colors. Then this is me marking a bat. But these 2 bats have a really interesting story. We set up our bat houses this year, and we actually marked this as our first bat by the bat house. So we named this one Pink Mohawk. We're not actually allowed to name our bats because it would go bias against our research. So we had to name it kind of what it was based off of, so Pink Mohawk. This one we actually just named Orange Dot because I put an orange dot on it. It's not in this picture, but orange dot. But we thought these were the same bats roosting there because, as you can see, they're pretty much in the general area. But we've looked further into it, and this is a big brown that has a darker pattern, and this is a little brown. And you can even tell by the bricks that they're different sizes. And it's really cool because they kind of rotated every night, and they're roosting in the same spot. And this is the first time we've seen this, so it's really, really interesting.
Our hypothesis in our research project was the number of bats in the Erie area would decrease from the previous census due to the presence of white nose syndrome. And here are some pictures of where on the route that we would go on at sunrise. This is a picture of an area at Gannon University called Old Main, and it kind of simulates the conditions of a cave. There are grates in the cave, and it creates like a secluded and dark area that bats like to roost in. And here are 2 other prominent sites that we would visit on this sunrise walk called East Pack and East Com Arts. And they're both, again, they're dark areas. There's a bat box at East Pack and East Com Arts. And the bats like to roost on the brick lining of the buildings. And so it makes a good area to find bats roosting. And the three most prevalent sites that we would find bats are, again, the Old Main site. We have South and North Old Main, and East Comarts, and our EPAC building. And those are the sites where we would most commonly find bats roosting.
So marked versus unmarked bats is how we collected our data. So if it's unmarked, we were able to spot the bat, but we aren't able to mark it. So that's where that comes in hand. The main important part of this slide is the years and how many bats. So this says 870 unmarked bats, and this says 395 marked bats back in 2012. Then we leap over into 2013, we have 94 unmarked and then 48 marked bats. That's a huge decrease into the bat numbers that we're seeing around campus. So we're starting to think that this is the year where white nose syndrome has impacted Erie, Pennsylvania. And then we skipped all the way over to 2020 because we had the same numbers throughout those years. And then you can see in 2020 we only had 7 bats. 2021 we only had 2. 2022 we had 5, and 2023 we had 6. And then this was this year's. We actually had 8 bats, which is a really good sign. So you can see we were able to mark, they were unmarked 5 bats and 3 marked bats.
Here are our pie charts of the distribution of bat species that we've found throughout the years. You can see there's a really prominent distribution of little brown bats earlier in the research project, but I feel like our diversity has grown over the past years that we've done this study. And just last year we have an even distribution of 4 different species of brown bats that we found. And this is a pie chart from 2024, this past semester. We found 8 bats in total, a majority of which were little brown bat species. We did find 1 silver bat and 1 big brown bat.
So the most important thing out of this slide is like the months. So you can see around this area we would start our bat walks in the spring where the bats were waking up out of hibernation or they're migrating back up north. And we have this huge incline in the summertime because the pups, the baby bats, are starting to leave the roosting sites of their mothers. So now they're trying to go on their own, and then we have this huge decrease in the fall time because now they're starting to leave for that migration route or they're getting ready for hibernation. So that's why we have these big numbers. And that's when we do our walks, is in the spring. Sometimes we do it also in the summertime to just kind of see what's going on, and then we definitely do it in the fall. We just ended it about like 2 weeks ago because it's starting to get warmer out. So we wanted to see if we would see any more, but we haven't seen any bats since last month. And then throughout the years, you can still see those steady inclines and declines throughout the years. And this was 2024. We found a bat in January, which is kind of weird, but that's why we do our bat walks like in various months because there's always got to be that one animal, you know. But you can see that incline and then the decline and the incline and the decline throughout the months.
A gist of our findings is that the bat population has decreased from 2010 significantly from numbers in the thousands to just 8 this past year in 2024.
So our bats making a comeback. They are making a slight comeback in Pennsylvania, especially in Bucks County, Knox-Nixon State Park. Michigan State University discovers that bats are actually resisting WNS because of evolution. So they're saying the heavier the bats are, the more able they are able to get off of the fungus, like off them. So if they're waking up with the fungus and scratching themselves up, if they have much more bigger body mass, they will not be so hungry to go looking for insects and leave that cage. And then because they're bigger body mass, they're able to keep their temperature in more. And bats found roosting at alternative temperature sites have also been observed that they might be making a comeback, so the warmer it is, the better they all are. This WNS is a cold living fungus, so it doesn't like warmth. Future plans, we intend to continue our progress through the 2025 academic year. We are officially a bat-friendly campus. We have bat houses on 4 of our buildings, and we have a ton more to put on. So we want to see if different sizes, different textures, even different colors will impact the bats. So we plan on putting more up, and so far it's working. They weren't in the bat house, but they were utilizing it. So that's a great sign. But our goal is to bring back the bats. We'd like to acknowledge Dr. Steve Borowski. He brought us onto this research, and I've loved it for the past 3 years. And we want to thank the Indiana University Biology Department for funding us, and all of our student researchers. But I want to thank you guys, because you guys are absorbing our knowledge about bats, and we want you guys to spread that.
That's the main takeaway about this presentation. So, thank you. Our sources. Questions?
So since they're seeing that those with bigger body mass are able to fight it off a little bit better, does that correlate then to certain species like the little brown bat automatically being smaller than a big brown bat?
Yes, so
Okay.
I've seen some research that yes, because they are smaller, they're not able to fight it off as more with the body weight, but because it is like more of a body weight to that specific species, that they are able to fight it off a little bit better.
I wondered, you mentioned seeing the little brown bat and the big brown bat in the same roosting site. Do you think that could signify potentially a lack of habitat? The two species are roosting together?
Yes, so because of downtown Erie, it's more like a popular bar area now, so at night there's more lights, there's more noises, so the bats don't really have an area, safety area to actually be hibernating. So having these bat houses on campus is actually giving them a nice secluded area, so if we have more bat houses, we're hoping that more like stay in there to sleep during the day. And that's why I also want to go into like downtown, maybe city council, to see if we can get some bat houses downtown to bring back the population in a more diverse area. So we live next to the lake, so there's so many mosquitoes, so if we have more bats, it goes into that trifecta. So hopefully I need to work with city council to help with that too.
That's awesome.
Yeah. Yes.
So that you have the mountains and bat-friendly campus, do you have like things posted so people know this is the bat house, like, you know, stay away, or kind of warned, because of the other things people can get from,
Yeah, so we wanted to see this semester if the bat houses would work, which it did, so we're really excited about. So we're probably going to do like a little QR code or something to like let the public know like what it is, or maybe a little poster. But QR codes are kind of big now, so but we definitely want to let the public know, and then all of our researchers go around talking to other professors so they know what they are. And we're also getting our clubs in our campus to get involved, so our environmental clubs are the ones who actually pay the bat houses. So.
Is Gannon doing any landscaping to attract bats, like white, yellow, flowering native plants, bringing moss or bigger?
So yes, Gannon is starting to go into that environmental friendly state. I believe we're getting a tree farm, or we're trying to, but we're trying to get Gannon to be more environmentally like inclined, because it is downtown, so it's kind of hard. But we definitely are trying to bring those factors into on Gannon campus. Yes.
So, you stated previously that you're not allowed to name the bats?
Yes.
Does naming introduce bias because it grows attachment?
Yes. So it's actually been studied that if you name the bat, or you name any animal of sorts, you become attached to it, because what if that bat, or what if another bat like attacked you, and you're like, oh no, that's Scar from like the Lion King, so you don't like that bat, so you don't want to pay attention to it. Is it a Simba? So that's why we kind of just name it on like the way we did, so like pink mohawk just looked like a pink mohawk, so that way there's no bias in the research at all. Yes.
I know this is like mostly the bachelor guy from Gannon, the Berkeley area, but you see, it was like mostly a population of the little brown bat, but you said the WNS is like easier, like to, I guess, kill the little brown bat. But do you, like, how do you, like, I'm kind of confused, like, how do you think, why do you think there is more little brown bats than like the big ones, since the WNS is killed?
Yeah, so for this area alone, we have a really big, like, high big and little brown bat area, like, population going on in Erie, Pennsylvania, because they're just so used to being around here. Like the silver-haired bat, we actually don't see. We were just really shocked to find it this year. So, because their body mass is smaller, doesn't mean it's harder to fight off. There are, like, researchers are starting to see that they are putting on more body mass. So even though they're still small, that little body mass that they put on might be able to fight off with it. And that's the climate change is also impacting that too. Does that make a little bit of sense?
Yeah.
Okay, thank you. Yes.
When the bat population was much higher in 2010, 2011, that kind of stuff, what were like the main species we had?
Yeah, so the main species we were having like this little brown, big brown, and then we had like a red colored bat, a white haired bat, but it was mainly the big and little brown because they're so populated in this area. This is kind of like their home state in a way.
Thank you very much.
Our next couple of presentations are going to focus on the eclipse that happened earlier this year. So Chris and Katrina are going to talk about how this affected animals at the zoo.
So my name is Katrina Orange, and this is Chase Bell, and this is our behavioral analysis of the animals from the April 8, 2024 eclipse at the Erie Zoo. When we started our research, we began with presenting the question of how would the results of the solar eclipse, including light, darkness, temperature, weather changes, etc., at a non-circadian time affect and alter the animal's behavior. So just kind of what an eclipse is before we begin. Solar eclipse is when the sun, the moon, and the earth all align and completely block out the light from penetrating. The eclipse passed through 15 different states. It started down in Texas and went all the way up through the northeast, up through Maine. And the eclipse was about 4 minutes long, but in Erie we experienced 2 minutes of totality.
During our study, we looked at 3 different species at the Erie Zoo. The first one is the Grant's zebra. These are from Africa and exhibit herding behavior there. We also looked at the Allen's swamp monkey, which are also from Africa. And then one more primate, the lesser spot-nosed guinan, which is from Africa as well.
So when we got to the zoo to set up our cameras, we made sure that we used Bushnell and other hunting cameras to collect photo and video of our animals. And we set the cameras up ourselves, but the zoo staff, we asked them if they could turn them on the day before and then shut them off the day after the eclipse. When we were setting up our cameras, we set them up about 5 days before to get the animals habituated to them, just to that new device in their environment. And we installed 4 cameras total. 2 of them were in the zebra enclosure, 1 inside and 1 outside, as well as we set up 2 cameras at the primate exhibit. That's us setting one of them up at 2 different angles to capture as much footage as we can, but we made sure that we kept them out of reach of the animals and then any people that were visiting the zoo on the days that we had them up.
So a quick analysis of our results. I first wanted to preview how it looks at the Erie Zoo. So there's 1 enclosure for the zebras, and there's 2 females there, an older female and a younger female. And then they have a primate enclosure too, which is where our other 2 species were at, along with a couple other primates. So first for the zebras, the older female seemed to stay inside during the eclipse, and the younger one was outside, seemed less influenced, and it was actually just doing laps in the enclosure. These are some trail cam pictures. I'm not sure if you guys have looked at them before, but they have a little bit of information about the date and the time here. And then you can see the ones that are in black and white were taken at night. So the first one on the upper left is at 3:15 p.m. This is just before totality. You can see that the younger zebra is outside, and you can just make out the older zebra's head already inside the enclosure. Then we go to the eclipse time. This is 3:17, so right after totality. The older zebra is still staying inside, and the younger one has started doing laps. And then further on, this is 3:19, and then 3:20. So further on during totality, you can see they're exhibiting the same behavior throughout, where the older one's staying inside, and the younger one is outside moving around a lot more. These are our primate results. So our first species was the Allen's swamp monkey. Over here you can see at 3:17, this is right after totality, this Allen's swamp monkey right here is moving into the enclosure. There's a door right here that you just can't see off camera. And then after the eclipse at 3:22, as it starts to get lighter, you can see it started to come back out. Our other species was the spot-nosed guinan, seen here. You can see this is during the eclipse at 3:19 p.m. and then after the eclipse at 3:22. The spot-nosed guinan seemed to be less affected by the eclipse and really just stayed outside the entire time. This is a bonus result with a species that wasn't originally part of our study. It's the black and white colobus monkey. We observed some pair bonding behavior, or at least what we think is pair bonding behavior, between 2 colobus monkeys and also 2 spot-nosed guinans. Hopefully this plays. So you can see what we think might be a mating attempt right here with the colobus monkey. And then they follow up with some grooming behavior, which is often seen like picking parasites off of each other. And then you might have seen in the back here, these are our spot-nosed guenons exhibiting a similar behavior, looking like they're picking off parasites or just interacting like that. We did have some inconclusive data. We also observed red pandas and lions at the zoo, but these two we weren't able to get data to include for the eclipse. The pandas were fed a few hours before the eclipse and a few hours after, and it seems like they were pretty food motivated because they weren't out at any other times but when they got fed. And then the lions were not outside from 12 to 4 during the eclipse, so we just weren't able to include them in our study. And the zookeepers kept the same feeding routine on the day of the eclipse as previous dates to try to eliminate any variables. This is a cool lion interaction we got that I wanted to show you guys. Even though it's not part of our eclipse, you can see the time was at almost 11 a.m., so that was before the eclipse happened, but it's pretty cool. So over here you can see the older female does a quick vocalization, and the younger one comes out here. Just kind of a cool angle that you don't always get to see of lions this close up. And then they're kind of standing beside each other, and then eventually do a little bit of grooming behavior. A little bit of extra discussion. So I looked on the internet and found some previous research into eclipse and zoo research from South Carolina Zoo in 2017. What's interesting is during this eclipse, they observed some sudden breeding behavior, which I thought was interesting because of that first clip I showed you where we saw some possible breeding behavior or pair bonding between the sets of the monkeys. There was some other planned research for this eclipse, but the data hasn't come out yet. So once that does come out, we will include it in our research. Not quite sure when to expect that, but we'll keep looking in the next couple months and include it if we do this presentation again. The atmospheric data gives us insight that the eclipse is more than just light. You also see a temperature drop, you see pressure changes, and so the trail cameras were not precise enough to show us whether the behaviors were due to light or pressure or temperature, but light is the one that we approximated them with. Here's a quick graph. You can see this is time from the beginning of totality. So this would be 2 hours before totality and 2 hours after totality. On the left we have light level, which is in detector counts. You can see right before and after the total eclipse, you can see that little flash of light appear, and then there was a sharp plunge in the light level. This data is from Dr. Horn and Dr. Conklin at Gandhi University in the physics department, and it was recorded using this device here. Dr. Horn will be doing a talk in a couple minutes where he'll tell you more about that. Our final considerations. Trail cameras can miss things that would normally be caught if you were there in person because they're set on a delay. Because sometimes you have animals feeding in front of your trail camera, you don't want to fill up your SD card in the matter of hours. So it's set on usually like a 15 or 30-second delay, which is helpful to not get 1,000 pictures of the same animal, but it means we may have also missed if one of the primates ran inside and back outside within like a 15-minute period. So we just acknowledge that that may have happened.
We also want to consider about the lack of replication, since eclipses don't happen every day and we only were gifted and lucky enough to see one of them. We only had one shot to collect this data. So looking back, we probably could have done or will do different stuff moving forward, but we only had one shot. As well as the zookeepers, they fed the animals at the same times that they always do, and the zoo was open to the public on the day of the eclipse. So there was also interactions with the visitors that were there. Some conclusions that we can draw, just starting, the female zebras, they experienced their behavior based on their age. Like the older one was inside, a little more stagnant in the doorway, and the younger one was pacing outside in a circular pattern. As well as the primates kind of had a varied behavior. So the Allen swamp monkeys, they went inside when the eclipse started in totality. The guenon monkeys, as well as the colobus monkeys, experienced the pair bonding and potential mating behavior that we saw. And we also recognize that light is not the only factor that could have contributed to the animal behavior on April 8th, but also light and temperature and weather and different outside factors. We just want to give a little special thanks to Dr. Epstein and the Erie Zoo for letting us do our zoo research there, as well as Dr. Warren and Dr. Conklin from the physics department at Gannon for providing us with their atmospheric data.
Thank you guys for listening. Are there any questions?
Did you look at their behaviors before or after the eclipse, like not just during? So did the female zebra, older female zebra always hang out inside?
So we did have the cameras on the day before the eclipse, but there were some times when they were feeding them at the eclipse time, and so we weren't able to draw any data that like at 3:16 the previous day matched up with on the eclipse day. But the information about the older and the younger zebra we actually got from contacting the zoo. We wrote them and they told us that the younger one seems to be a little more energetic and the old one stays inside. But we didn't have any, conclusive data between the 2 days.
I also wondered, do you think that the animals being captive and essentially having light levels manipulated by being brought inside might have impacted the kind of lack of behavioral changes that you saw in some animals?
Yeah, definitely. I think, out in the wild, it's going to make a bigger difference because they're going to have maybe predators coming out at dawn and dusk when the light levels are changing, but those that have been bred and grown up in captivity aren't going to see those predators. And so, we were actually interested to see if they were going to change at all. We didn't know if they would just be habituated to light changes all the time from people turning on and off light switches and not do anything. So, I kind of wish we had maybe a wild example of this, but obviously there's no zebras here in North America, so don't have any of that data.
So during the 2017 eclipse, you said that at another zoo there was observation of mating behaviors and stuff. Do you know what species those were?
No, I didn't see any information on what species they were. I think it occurred in multiple species. So the report that I found just mentioned that there were a couple examples of that mating behavior. But I would assume that it would probably be in primates because that's where we saw it in. Sure.
Why did you pick these 3 species to focus on?
So we only picked these, we picked 4 from the beginning, but we didn't get video footage or photos from the other 2. But this is just what the zoo recommended and the different zoologists, because we kind of had an idea that we wanted to test these animals, but we didn't know, and after talking to the zoo more and the people in the Erie Zoo, they helped us come to these 4 animals.
Do you think that based off the animals' behaviors, for example, like the monkeys, say, breeding and stuff like that, did the solar eclipse, did they either know or did they think it was something else, something that triggered their, it's mating season, this, that,
Yeah, that's something we're not sure about. That would be really cool if we had, more sensory data. Maybe you could record heart rate and blood pressure, that type of stuff to determine whether they were changing things like they do when it gets dark at nighttime, or if it was, like you said, something triggered by maybe a shorter day length. They thought it was shorter than normal, might trigger something like mating season. So those are all cool variables, but something that unfortunately was not part of our study.
Do you plan on expanding your research in the future following different eclipse patterns and stuff?
We would love to. Unfortunately, it's going to be a while before there's another North American solar eclipse. But it'd be awesome if we could do it in another country. Or, like I said, as more data comes out from this eclipse from other zoos that were in totality, we could try to do some larger analysis of those zoos combined with our data to get a more rigorous example of what happened.
Thank you very much.
Or you can use the computer.
All right, our next presenter, also from Gannon University, Dr. David Horn, is going to talk about life coaching.
Excuse my voice a little. I'm getting over a fairly heavy cold, so hope everyone can hear me. So we're continuing with the eclipse theme here. In this case, we're looking more at the atmospheric effects of the eclipse and on a nationwide scale. Before I get into that, I'm not showing pictures of myself out of some kind of egotism. Well, not completely anyway. But I'd just like to mention that this particular facility here at Presque here, this is me giving a talk here on that monitor over there. And I gave a lot of talks here, so I really appreciated the support from this facility. And also some of the work that you're going to see here was actually supported by volunteers at least one volunteer that carried one of these units to Texas for me, and I wouldn't have made that particular connection had I not presented here last year at this time. So I really appreciate what came from that. We did a lot of things for the eclipse. I'm not going to go into them all. We built great big solar filters and things like that. If anyone's interested in those, I'm happy to talk about those. We also gave out a lot of glasses. But the focus today is really looking at what happened to the atmosphere during the eclipse and how we measured that, and really getting into talking about that a little more. As you heard in the previous talk, if you're waiting for the next eclipse here in Erie, you're a little out of luck. That's not 2144 this evening, that is the year 2144, when you'll have to wait for the next eclipse. This is actually the conditions here from downtown Erie here. So this is the McConnell Stadium at Gannon University. And if you were here witnessing this eclipse, you were along this line of totality, which is pretty much what I'm going to talk about. So here in Erie, you could see we were pretty much the only major city in Pennsylvania that got the total eclipse, hence the excitement around it. Much of the rest of the state did end up with partial eclipses, but what we really wanted to do was study what is the effect of this particular shadow as it races across the landscape. So this is not the North American eclipse, this is over Indonesia, but it does show the size of the shadow relative to the planet itself. So the actual darkest part, the umbra that we refer to, is really quite a small spot. And it's the head of this cone that's coming from the shadow of the moon. And the contrast against the water there just looks really nice, so I just wanted to show that. And it's the fact that you have this very localized shadow that's moving with some variations in speed, but at about 1,000 miles per hour or so across the landscape that we wanted to look at. How does that affect the weather, and how does that affect general conditions of the atmosphere as we are experiencing all of these various types of illumination over a couple of hours? So essentially we're seeing the illumination level going from a full sun to completely blocked out just over about an hour or so. So what we really wanted to do was say, are there any atmospheric effects of the eclipse? And if so, what are they? There has been reports previously about an effect that's called the eclipse wind. Some people say this existed, some people say it didn't exist. So that was a bit of a discussion. Some actual studies found it, some didn't. So it's a bit inconclusive as to whether a breeze is set up during the eclipse. And most of it is people saying, oh, I experienced a breeze. Did they experience a breeze, or did they experience a drastic drop in temperature and just their senses told them it was a breeze? Was a bit of a question. So that was what we really wanted to answer. And people have said, well, why don't you just pull data from airports and things like that, from existing weather stations? That kind of works, and it does actually support some of our work, but they're not calibrated all the same. So what we wanted to do was take a series of calibrated anemometers to measure a whole pile of different atmospheric variables that I'll mention in a second here, and just place them across the country, all built and calibrated in the lab, and then we could actually see as the shadow races across the entire nation and getting some really good coverage all the way across the continent, we could then see what happens in terms of the pressure, in terms of the light levels, in terms of the temperature. and then we could actually measure those effects from calibrated sources. That also meant, though, having calibrated anemometer equipment. So we built these things. The centerpiece of these is actually a quite expensive sonic anemometer. There's our lab. There's coffee in the corner that was essential to me, at least. Dr. Nick Conklin did most of the electrical engineering on this. I attached them to tripods and designed them. But this device is at the front if anyone's interested in taking a look at it. Dr. Conklin, in his usual way, came up with the acronym SEWA for these, which I didn't like, so I changed it to Total Eclipse Atmospheric Sensor Experiment, TEAS, which is a little more professional in my opinion. We were going to build 6, but we actually got the money for 7. What's in these? Well, there's waterproof casing, the best that Harbour Freight could provide. We've got a sonic anemometer there that records most of our data. That is the centerpiece of this equipment. It does wind speed, wind direction, pressure, humidity, temperature. We also had a secondary pressure sensor just in case there was a problem with the main anemometer, so we would get some data. And we also had a light sensor, and you saw some data in the previous talk from that light sensor for the Pennsylvania sites. Where the connections I made here really helped was in terms of research participation. We did want to have 6 or 7 sites across the country here where we wanted people to operate these. So it was really simple participation for anyone involved. They would either receive the anemometer package in the mail, or I'd hand it to them and they'd travel with it. They had to be on the line of totality, otherwise this wasn't going to work. All you had to do was point the unit north, and then an hour before first contact, switch it on, and an hour after last contact, when the sun's completely back in the sky and the moon has moved completely off of it, you switch it off. And then you repackage it and you ship it back to us. And that was all you had to do. A couple of sites, people were annoyed because they wanted more to do. They wanted to play with knobs and press buttons and things like that. But the general idea was to make this as simple as possible so there weren't going to be any mistakes. Didn't quite work. So there's what you get. You get a compass, so you knew which way was north, some instructions, and a letter that says, please TSA, don't blow this up on the tarmac, it's not a bomb, and a tripod so that you could actually mount. Taking these on site was interesting. I'm just going to mention this because I can't help myself with this part. So we also took our telescopes out on site and our anemometers, and we were going to various places to test these equipment. While we were there, most of the public that we engaged were really interested, and we gave them eclipse glasses, spoke about the eclipse. A few people were a little suspicious of our activities, wondering what we were doing with this stuff. And so I wound up putting hazard tape and signs all over my truck so that we could give us some legitimacy. And it's amazing that you can have as many degrees in physics as you like, but nothing gives you legitimacy like having a truck with a sign on the side. In terms of the eclipse, we did have an annular eclipse, and this is what I reported on here last year, which gave us a great opportunity to test the equipment. So we tested it here from Pennsylvania, simultaneously from San Antonio, Texas. And as you can see, Pennsylvania didn't let us down that particular day. There's rain all over the device there, which was great from a testing point of view, because it really meant that this was a resilient device and we could put them up in any weather. I'm not going to dwell on the partial eclipse data. It was partial, and it was cloudy, and it was windy and raining, so not a lot from Erie, but we did get some good responses from the San Antonio site, which actually does help us with the total eclipse. Make sure we could interpret those results nicely. In terms of the research, this was basically, in one case, a cold call. In some cases, people we knew, other people I'd previously collaborated with. We ended up with the following sites. We ended up with Texas, Arkansas, Missouri, Indiana, Pennsylvania. We had 2 sites because we wanted to make sure that our home site had a calibration, so we could have 2 exact devices working. One was in Mill Creek, one was on the Gannon campus with me. And we ended up with Brockport, New York. That particular group was supposed to go to Canada, but one of their group had an expired passport and they wouldn't let them in, so they went to New York. Out of all of these sites, we managed to get great data from every last one, apart from the Arkansas site, where the system went into a safe mode and they didn't read the instructions, and so it didn't work. Also, one of these pictures is not like the others. These are supposed to be on grass for thermal purposes. This one is not. So one site, not picking on them, didn't do exactly what we should, and it was user error that resulted in that not coming back. But I think we got a reasonable return for our investment and for our volunteers. So I'm going to show some results here. Bear in mind that we're looking at 6 variables here over 7 sites, and we're trying to look at the best way of smoothing this data and correlating it between each one of these sites. And I came up with lots of ways to sort of present this quickly, and then I thought, forget it, we'll just look at the actual raw data. And if we can see changes in the raw data, then once we process it, those should become even more apparent. So bear with me with some of the noise in some of these, but you're going to see some results, especially on things like wind speed, where this is recording at millisecond durations. So you've got quite a lot of noise in some of these, but they won't stay like that. First of all, as you saw in the previous talk, we do have a light detector on this particular device. That light detector really saturates in the sunlight, and then during the eclipse basically reaches a value of 0. That's really useful for us just from the perspective of getting individual times for when the eclipse hit each site. So we can trust, the calculations that have been published, but this gives us the exact time and gives us the ability to find where was the maximum, where was the middle of the totality for every one of our devices. This is the only plot that I'm going to present in universal time that's gone kind of off the screen, but this is universal time, and what this is actually showing is the eclipse as it hit every single one of these devices across the country. And you can see the 2 Pennsylvania sites are pretty coincident. The New York site is much wider. New York was our only cloudy site. Now, Pennsylvania did have some cloud, but we still saw the eclipse. As people who live here know, we saw the eclipse just about. It was a dodgy thing whether we would. So the New York site does have a wider light drop than the rest of the sites. What we were looking for was really changes in various atmospheric parameters. So here is the actual temperature. The way I'm presenting this is I'm showing this in terms of normalized time. So we've got 0 here when the totality occurs, or maximum totality occurs, and then negative is before the eclipse, positive is after the eclipse. Different sites turn their units on at different times in some cases. And so we have different sort of temporal coverage. You can see the Texas site, which is the grey one up here, they turn theirs on 1 hour before the totality rather than 1 hour before the actual first contact that occurred at the very beginning of the eclipse. So there's a bit of change. If you look at these, what we're really looking for, like I said, is change, and hopefully consistent change from each site. Unfortunately, the Pennsylvania site that I was actually operating, and I'm sure people sort of corroborate this, had some interesting weather going on. Probably a cold front moving in as the actual eclipse was occurring. I remember an actual very significant temperature drop that I felt. So it's a bit of a mess there showing all of them together, so I'll just sort of take it down a notch here. This is essentially for our northern sites, and the temperature contrast that we're showing there is based on the temperatures about 2 hours before the eclipse, actually before the 1 hour before the first contact, down to the absolute minimum temperature. So this may not be the final temperatures that get reported, but I know for a fact, being at the PA1 site there, I felt the most significant temperature drop I've ever felt out of 3 total solar eclipses. It was quite prominent. There's also a bit of a mess here in our Pennsylvania data because it tended to get cloudy right after that eclipse and it stayed cloudy, whereas the other sites didn't tend to experience that. But there's a fair consistency and a drop in the temperature between the New York and Pennsylvania 2 sites, the Mill Creek site. What is more consistent is Texas is a little noisy, But Missouri and Indiana, I can definitely see the change here occurring around the totality and then really reaching sort of minimum temperature here around 10 minutes after the total eclipse actually occurred. So, like I say, there's some value in looking at this raw data here, and we can actually see those drops consistently, and I think it's there in the Texas site to a lesser degree, but Missouri and Indiana definitely have similar temperature drops and similar form to those temperature drops, which is interesting. We also have humidity data. Not going to bore everyone to death with lots and lots of graphs, but our humidity, again, Pennsylvania, a little odd, but it does coincide with some of the patterns we do see in the New York data. So disentangling these is really the current task that we're involved in, trying to cross-correlate these things and see if we can find these relative changes. But I'm definitely seeing that they are there. Humidity, even during the annular eclipse, undergoes a serious drop during the eclipse, during the total eclipse. I think there's a correlation in that one, though. I'm quite happy with that one. So this is the Texas, Indiana, Missouri sites here in these various colors, and I think we can definitely say, without much doubt, that there is a humidity change during the totality right there, and leading up to and afterwards. Pressure, again, we looked at our pressure responses. Pennsylvania, nice and self-consistent, but again, the Pennsylvania sites with their individual weather probably aren't giving us a great gauge of what's actually happening due to the weather effects of the eclipse. But when we take out Pennsylvania, the rest of them actually do follow a very similar trend. They are probably going downwards just as the general diurnal trend during the day, but there are drops here associated with the eclipse in most cases. Indiana gets a little strange, but most of them have very similar form to them. So hopefully once we've actually processed all of this data, we should be able to see a little more in terms of whether we have consistent changes from one site to the next. Wind speeds, we expected to see a change. We did get a change. We got drops in the wind speeds. Wind speed is a lot noisier due to the nature of the sonic anemometer, as you can see. So we've kind of got a work cut out for us in terms of smoothing, but not destroying our data here and not accidentally fudging this to make it look like something that we expect to see. This a little more consistent in the Indiana through Texas sites once again. Wind direction, which is one of the big things about the setup with the breeze, is one of those things that is quite difficult to actually take into account. We can produce these nice windrows diagrams, which will tell us at what point the wind was actually shifting. And when we do that in nice increments, before and after the eclipse, we can actually see a shifting of the wind and then a return to the wind's original direction in about 4 of our sites. But correlating the wind direction is difficult because you're looking at something that goes from 0 to essentially 360 degrees, and you're trying to convey that on a diagram. So we are currently looking into taking standard deviation of all those sites and attempting to plot those together. Sort of the early days in how we actually express this. If we do that, we do see a change in most of our sites coincident with the eclipse itself, or just after the eclipse, around 15 to 20 minutes afterwards in terms of wind direction. It's hard to tell from these graphs, and like I say, I think Pennsylvania 1 there, which was the Gannon site, is pulling us off a little bit. But I won't go into too much detail on those. I think that we can definitely say that we have seen changes in the temperature, the pressure, the humidity and the wind speed coinciding with the total eclipse and the annular previous to that. Wind direction, that's going to take a lot more work. I wouldn't like to make a conclusion here about whether that wind direction shift was genuinely to do with the eclipse just yet. I think we might find some slight correlation there. But that's currently what we're working on. There is a paper actually that's been submitted that I'm waiting on to finally work on to do the last changes, so hopefully that will come out soon. But these units performed really well. We're really quite pleased with how they performed. And they gave people, faculty, students across the country the opportunity to engage in some research that didn't ruin their eclipse experience, because they could switch it on and then just get back onto looking at the spectacle of the eclipse. Future work here, well, we want to carry on doing this, and this is why I want to get some of the results out there. We would like to make the light sensor a little more complex, a little more of a photometer rather than an actual just light sensor. But it would be nice to actually renovate these and make them a little more robust in some ways for use in the 2026 eclipse here, which is actually occurring in Greenland, Iceland and Spain. And once again, do a very similar thing. Find partner groups in these locations and place these anemometers along the line of totality once again and see if we can get some data there. The great part about the North American eclipse was it was all across the land, it was all across the continent. Here we do have the ocean in the way, so we do have to get a little more diverse in who we actually contact to help out in these cases. I do want to throw some acknowledgements out there. Jake at the University of Missouri, Joyce here, who took one device to Texas for me, and a few other people, University of Arkansas, Yamaha, and NASA GSFC there. And shout out to the people who actually funded this and allowed me to buy this equipment in the first place, basically being NASA and Gannon. So that was a very quick run through of the kind of data that we have. There's more to come on this, much more in-depth data analysis, and I'll just show this last image of the eclipse here just to finish on if there's any time for questions.
Time for a question?
I'll go from back to front. So.
I didn't see Arkansas on any of the data. Was that because
So what happened was the Arkansas people, this goes into a safe mode sometimes, and in the instruction manual it said if the light comes on to tell you it's in safe mode, you've got the incredibly complicated procedure of switching it off, then switching it back on again, and then taking it out of safe mode. They didn't do that. They just flicked the switch and put it up, and then they didn't look at any of the lights. So the only things we got from Arkansas were the secondary sensors. We got the light level, and we got the secondary pressure sensor, but this anemometer was in safe mode. So we got all of the other sites, we just didn't get a complete data set for Arkansas. Go.
So just quick questions about the PA site specifically.
Mm-hmm.
Did you put any standardization into what the surrounding area around your devices had to be? Like if it was, say, in a city with a lot of tall buildings that might change wind, or like the proximity to the lake when you're at the Gammon site versus wherever your Mill Creek site was? Because Mill Creek's could be close to the lake, or it could be much farther.
Proximity to the lake is something I seriously considered in terms of the effects that we were seeing in the PA1 site there, because PA1 was the Gannon site. You're right on the lake, and you're going to get different convective effects there based on the fact you've got a huge body of water and you're right next to it. So once again, this is why the analysis is taking quite a while, because I'm trying to read into that and see, is it due to some kind of convective effect? And maybe the fact that the lake is a big heat sink right next to us that gave us that weird response. Or was it the weather? Because unfortunately the weather here didn't cooperate as well as we wanted. And at one point I thought we were going to completely miss that eclipse. So it's going to have big error bars on Pennsylvania too, but it is something I am seriously considering. And I'm going to run some other tests with this on different days to see if we can sort of see that kind of pressure variation based on just running it on a standard day.
So going back to the Arkansas data, I know that light is one of the few collected things. The light exposure seemed to be just a little bit more for the Arkansas data, and it was consistent throughout all the other cities. Was that due to, I guess, the various human errors in Arkansas, whether it be the concrete or like what happened,
it's pretty sharp-eyed because I noticed that and I didn't like that. That particular light sensor has a slightly different response to the other light sensors, so it doesn't actually go all the way down to 0. So that's just a quirk of that particular piece of equipment. I did notice that, and when during testing, because we tested these in the lab, we put these on tanks so they're all at the same pressure, and that one always gave a very slightly elevated light level. But we weren't really too worried about it because the light level is really there to say saturated in the sunlight, minimum value, and then give us that timing. So yeah, it is slightly different.
Dr. Horn, it'd be interesting if you played the audio during this little segment right here where
Yeah.
you're extremely excited and mentioned a few.
There's there wasn't an expletive. It wasn't that bad. I did not expect to see quite as active a sun during this particular eclipse. Out of the 3 total eclipses that I've seen, this was the most active of the 3. So yeah, I was a little bit on the excited side. There is this is a director's cut version, basically. There is another version out there where the audio gets a little spicier, but not too bad. It's still kid-friendly, just about.
And the other question is, do you have any connections with zoos in Greenland, Iceland, or Spain that we can go to in 2026 with you?
I don't, but what I'm hoping to do once this data is out and trying to get this published is we could send people that and say, look, we've been doing these studies. We could send one of these to the actual zoo. You could do your animal thing right there and have a qualified operator switching these on and off to make sure they don't go into safe mode. So I'm all for that. Any eclipse work that can be done, always interested.
Thank you so much.
All right, we are going into our lunch break right now. Everybody is invited to just go right across the hallway lobby into the cafe. There's lots of lunches there. You can sit in the cafe and eat. You can bring it back here and eat, or if you have to run for some reason, you can take it with you. But I am excited about the next session, which will start at noon, and that will be our fisheries session. So please come back. We'll see you then. Thank you.
Yo.
each hand.
I got really fun videos. Yeah.
Yeah.
All right, everyone, we're going to get started with our second session of the day. So to start us off here, we have Mark Hafley from the Fish and Boat Commission, who will tell us about the status of the Lake Erie fishery. Mark?
Thank you very much.
Yep.
All right, so good afternoon, everyone. My name is Mark Hafley. I'm a biologist with the Pennsylvania Fish and Boat Commission's Lake Erie Research Unit here in Fairview. And we're going to talk a little bit about how we come up with our data, some of the catch rates for the fishery here in Pennsylvania. But before we do all of that, I just want to say that we have one of the absolute best people who performs our krill survey for us every year. He's outstanding, and without his help, we wouldn't be able to get all this wonderful data that we get. So, and he's sitting here. Thank you, Mark Letheby, for all of your hard work, for all of this information. So the way we figure out basically what's harvested from Lake Erie every year is we have a bus route style krill survey. We operated at 4 sites, Walnut Creek Marina, Northeast Marina, Lampe, and East Avenue boat launch. From last year, what happens is as Mark goes to these locations, he counts the boats that launch, he counts the boats that land. Of the boats that land, he interviews those anglers, gets an idea of what they've caught, what they've released, and then we can extrapolate that out based on how many anglers per boat, how many boats launch. We can figure out how many boats launch out of those 4 sites or in the Erie region throughout the summer. So for last year, the 2023 data, we estimated that roughly 27,840 boats launched here in Erie to target fishing in Lake Erie. So we averaged just over 2 anglers per boat, 2.5 anglers per boat roughly. And then from that, we're able to figure out how many fish, how many hours of effort. So along with all of that, throughout the week, we go down to the gut houses at Walnut Creek and Northeast Marina. We cut carcasses and we can get an idea of the fish that are coming in, the age classes for exploitation, sex ratios, and size range of the fish that are being caught. So from doing all that, estimating how many boats, how many people, how many fish are caught, and what age classes are caught, we're able to come up with catch rates. We're able to come up with how many pounds of fish are harvested out of the Pennsylvania portion of Lake Erie every year. So Erie is just a phenomenal fishery. It's highly prized. It's highly targeted. As you can see, with almost 229,000 hours of angling effort directed at fishing here in the Erie region. So it's big business for us. So the king of Erie will always be walleye, and walleye is one of the most strictly managed fish. Walleye and perch are the 2 that we manage the hardest here in Lake Erie with all the jurisdictions. Walleye and perch are the 2 main commercial fisheries that come out of Lake Erie. Pennsylvania is the only state that allows for any commercial harvest of walleye. Ohio, Michigan, and New York do not. And then Ontario has a pretty hardy quota of walleye that they harvest commercially every year. I think this last year was almost 12 million fish they were allowed to harvest commercially for walleye. So we're currently in a period of high recruitment. There's lots and lots of walleye out there. There's lots of happy anglers out there. And you can see by the graph on the bottom that it's continuing to trend upward, which is good for fishing. It's good for business. And in a situation like this, one of the limiting factors would be food. And we were worried about the forage base within Lake Erie. But one of the things we've noticed last year's trawl data from October, some of the highest forage rates that we've ever seen in Lake Erie. So even with all these mouths to feed, there's lots and lots of food out there for them to eat. So as we've seen the eutrophication of the Western Basin, we see the warming up of the Toledo area and off the island in Sandusky, we've seen a shift in walleye production into the Eastern Central Basin and East Central Basin. So we're seeing a lot more fish born down here. And the East Basin and the Central Basin of Lake Erie is much more conducive to walleye reproduction and walleye life cycles. So they're doing very, very well now that we've seen that big shift. So there was a huge year class in 2003 that drove the fishery until the big year class of 2015, and then it's been pretty consistent since 2015 with the year classes. So the majority of our anglers targeted walleye. We estimated almost 424,000 walleye were caught out of Lake Erie last year and harvested almost 240,000. Again, just an incredible amount of fish. Back in the late 90s and early 2000s, in this period through here, we typically harvested about 80,000. So just to kind of put it into perspective, our typical harvest every year was around 80,000, and last year was 239 plus. So with that population estimate of 2023 with the 93.6 million fish, just a ton of fish out there, and lots of happy anglers. So when we look at the work that Mark has done and the Krill survey information, just over 87% of our trips were targeted at walleye. So all the interviews that we gain every summer, just over 87% of them were folks that were using directed effort for walleye. So we asked them what they were fishing for. Some guys tell you they're fishing for anything, but 87% were looking for walleye. So catch per effort was almost 1.5 fish per hour, which is incredible. That's your limit of 6 fish, in a half a day. So it's just an incredible fishery. And then we're seeing a harvest of 0.8, and that's the total or the average harvest for the entire year. This year in July, we had a harvest rate of 1.4 or 1.04 fish per hour. So over 1 fish per hour. It's the second highest in the time series since we've been keeping track since 1993. So that's how good fishing has been. So yellow perch is a different story, and we would think that potentially it would be due to predator-prey relationships, but it's not. We are seeing that, again, with the walleye fishing being so good, we're seeing a decrease in effort directed towards yellow perch. We're seeing a population that's rebounding and coming back, as you can see in the last 3 years. We're showing a better population than we've had in the last few, but we're still showing terrible catch rates. And one of the reasons for that is we're seeing just folks not doing it. Currently, we had 155 million H2 plus yellow perch in the population for 2023. And we have Management Unit 3, which is this portion of Erie. So it's basically from the Ohio line to 6 Mile. And then we have a small portion of Management Unit 4, which would be from 6 Mile to the north. And almost 57 million of those fish, of the 155 million, reside in that Management Unit 3 section, which is right here. We saw a decline in the fishery and in the catch rates in 2015. And in 2015, we saw a huge increase in spiny water fleas in the diets of yellow perch. It completely reverted from fish and went to mostly spiny water fleas. So spiny water fleas are pelagic and up in the water column. Our anglers go out and target fish on the bottom using emerald shiners or some sort of minnow for bait, and they weren't interactive. They weren't fishing where the fish were, and the fish weren't eating what the anglers were using as their bait. So this last year, 1% of or in 2023, 1% of all of our interviews were targeted for yellow perch. But again, it's because of this huge walleye year class, the huge walleye fishery. So one of the things we notice is that our anglers are very smart, and fuel prices being what they are, they target something that's going to yield the best bang for their buck, if you will. So when walleye fishing is slow, guys will go out and they'll angle for perch, they'll anchor, and they'll jig. However, cleaning 6 walleye seems to be more desirable than cleaning 30 perch. And it's probably a give and take for amount of meat that comes out of those fish. So when walleye fishing is even remotely good, we see this huge shift from perch to walleye. The catch per effort, 0.3 fish per hour, with everything that was pretty much caught was harvested, same exact harvest rate. At the height of perch fishing back in the middle 2005 to 2012 era, it was almost 3.5 fish per hour, and last year it was 0.34 fish per hour. So it is a huge decline. We did have anglers that found them this fall and did really, really well. They targeted them. They caught a lot of perch. They were very, very happy. And then they wanted to know where they all came from, but they're all 12 to 13 inches long. They're 5-6-year-old fish. They've been there, just nobody's fished for them. So we are seeing some folks that are starting to come back to perch fishing, which is really good. So we also have our lake trout fishery, which is gaining popularity. We stock, it's all done through stocking out of the Allegheny hatchery. The U.S. Fish and Wildlife Service stocked these fish for us. And we stocked 140,000 fingerlings last year. And we really see this fishery starting to increase with our catch and release anglers. They're not much for table fare, but there is a trophy quality to them. There are very large fish out there. And you can see we had just over 4,000 hours of directed effort towards lake trout. They caught quite a few with over 2,000. And these fish range anywhere from 6 pounds to the biggest one we saw last year was almost 27 pounds. So the state record for lake trout is 31 pounds, but just over the line in New York, it's 40. So more than likely the next state record to fall probably will be the lake trout record. We see a large directed effort in the month of May. May seems to be really good. The fish are still near shore, eating for the summer, or putting back on weight after winter and after they spawn before they go into the offshore waters. But from those estimates, we're pretty satisfied that those estimates are correct. We did a voluntary charter boat study. We had a bunch of charter boats that, as they took their clients out, they kept track. So 242 hours and 54 trips, 582 caught, 234 harvested. So, if you look at those fish, the average length is 800 millimeters with the average weight of almost 11 pounds. So they are a really, really nice fish. They are, sought after by catch and release anglers. And the charters are starting to see a decline in their lake trout trips only because folks don't want to pay for a charter to throw fish back. They keep them, they eat them, and then they realize that they're not much for a food source, but they are a really good fish for catch and release. So, but it is a booming fishery. We used to stock at the East Avenue boat launch and at the Northeast Marina to try and spread them out. We know that we have a spawning aggregation off of East Avenue, and we have spawning aggregations off of 16 Mile. But we were noticing a lot of bird predation within those two marinas. The fish were essentially locked in there, couldn't get out. So we shifted the stocking these last few years to the Lawrence Park Marina, the Lawrence Park boat launch. There's no marina there. There's nothing to hold them in. The fish can disperse at their will. There is still some bird predation because they aren't the smartest of fish, and they do swim back up on the beach. They swim on the surface and the birds eat them. But by and large, a good portion of that 140,000 made it back. And we stocked another 80,000 this year. So we stocked cyclically lake trout and Lake Erie. So it's 140,000 in Pennsylvania and 80,000 in New York. And then it's 140,000 in New York and 80,000 in Ohio. And that's 140,000 in Ohio and 80,000 in Pennsylvania. So we're working on kind of the boom and bust reproduction system that we seem to see in lake trout to try and see if we can not only spread out the fish and get some natural reproduction, but also spread out the stockings and not stock as frequent and as small numbers in all three jurisdictions every year. So we have an open lake assessment of our steelhead fishery, which isn't as popular as our tributary fishery that's currently going on. But we do have folks that go out there and actively target steelhead through the summer. There's a couple tournaments in the middle of the summer that target steelhead, but mostly it's just incidental fishing throughout the summer. The majority of our targeted effort is in September and October when they're staging at the creek mouths before coming in. So again, 3,600 hours of effort directed at the open lake with 1,800 caught and 1,200 harvested. Those catch rates of 0.22 fish per hour have been pretty stable for the last 12 to 15 years. We've seen this very similar catch rates, which shows a very stable fishery, which is something we're very pleased with. There is, again, same thing, there is a trophy aspect to it. Guys are catching just wonderful fish this year. We haven't had any rain, so we haven't seen them, coming into the tributaries like they should. But with the October trawl survey last year and the overabundance, essentially, alewives, they just had a very good preferred food source, and we're seeing some of the biggest steelhead that we've seen in recent years being caught this year. So if we look at our tributary side, the Pennsylvania Fish and Boat Commission has always run a collective angler logbook. We mail you the logbook, you fill it out in pencil, you mail it back to us, and we crunch the numbers. Towards the end of the program, we had about 8 people participate. We didn't really get the information that we wanted. It was very kind of sad, but the catch rates were very similar. We could see the trend even with the low number of participants and the low number of actual trips. But then the Pennsylvania Steelhead Association started their own krill survey that is on the phone. You can log on to the app, you can plug in all your information, it does individual trips, and they just have a huge participation that we never had. So from our 8 to their 162 and our 40 trips to their 475 trips, it's a huge difference. However, it yielded the exact same catch rates at 0.7 fish per hour, whether it was our 8 anglers or their 162 anglers. We saw the same exact catch rates. So the difficult part about all of this is trying to convince folks that even though you caught nothing, zeros are still very important, and you still need to enter your zeros, which nobody does. So we make up some portion of those to make sure that the zero trips are accounted for, so that we don't overinflate our catch rates. But again, 1,900 hours of fishing, 1,300 caught, 115 harvested. It's a huge catch and release fishery, but it's even bigger amongst the folks that deal with the Steelhead Association. So again, trying to look at the bias that could be due to the anglers that participate, could show the bias in catch and harvest. But we still don't see a lot of harvest in our steelhead program. But one of the most important things that comes out of that survey, in my mind, because everybody wants to catch a lot of fish, everyone wants to catch all the fish, they want to catch them fast, they want to catch them close to their truck. So one of the questions that the Steelhead Association asks is satisfaction. Are you satisfied with the fishery? And overwhelmingly, completely satisfied at number 5. Very few down in the unsatisfied. So we know that our anglers are enjoying what's being done, enjoying the fishery as it is. As we're moving into the changes that the steelhead fishery is going to encounter into the future, we'll see how that all goes. I do know that we did have a diagnosis of VHS in the hatchery 2 years ago. The lab contacted the Department of Ag, Department of Ag contacted the U.S. Department of Ag, and they essentially came in and quarantined both hatcheries, Tynesta and Fairview. After we went through a period of 30 days through quarantine and destroying some of the fish, or some of the eggs from the tanks that tested positive for VHS, we found out from the lab it was a false positive. However, the USDA was already involved, and we're no longer allowed to raise steelhead at the Tynesta facility. So all the steelhead will now be raised at Fairview. Fairview is the smaller of the 2 hatcheries. They stock typically about 1/3 of the million fish that we stock. So we're currently in the process of doing a feasibility study, which will be done here in the next few months. And then we've allocated $25 million to building a new hatchery here locally to get back to that 1 million fish being stocked moving into the future. Now, with the time frame for that, who knows how long it will take to build such a facility. The other difficult thing is that, believe it or not, there's no water here. We've drilled 12 or 15 test wells, and the best well we found so far tested at 100 gallons a minute. So we need about 1,000 gallons a minute to supply this hatchery for all these fish. So we continue to look. We're invested and dedicated to this program, but it is changing. It is evolving, and as we go through steelhead season, we'll start having anglers noticing impact, and we'll see how these numbers change through the years. We're currently doing an otolith microchemistry study. We're extracting otoliths out of angler caught fish from all of our tributaries, and we're going to look at the proportion stocked and the proportion returned and try to assess, is there a difference in return rates or return proportions from one hatchery to the other, being that the Fairview fish were born here in the watershed and should have keyed in on those chemical signatures and therefore potentially don't stray as much as the Tynesta fish, which were born in the Allegheny watershed. But hopefully, by the middle of next summer, we will have results from that study, and we'll be able to better inform our stocking practices and how soon we need to get back to stocking 1 million fish. Presumably, the fish in Tynesta stray more, and the fish in Fairview do not. So we're hoping to see a higher portion of Fairview fish in the tributaries than Tynesta fish. So we know that those Fairview fish do well, and it might give us a little more ease of mind knowing that our catch rates will stay, probably not as high at 0.7 fish per hour, but in an acceptable range until we can get back to full production. So with that, I will ask if there are any questions. Yes.
So are the edible ones generally safe to eat, or do we have to worry about things like heavy metals, microplastics, that kind of stuff?
So Lake Erie has known levels of mercury and PCBs. So the fish from Lake Erie all fall under the consumption advisory of 2 meals a month. I haven't seen the results as of lately, but it all was dependent upon how the study was done, as far as I was aware. So if they ground up the whole fish, they did find those contaminants in the skin and the visceral and all that, or the visceral fat and all that. But when they just ground up and did it with just the filleted portion, it was very, very minor. So they are safe to eat, but they still fall under the consumption advisory of 2 meals per month. Lake trout, because they're such a long-lived fish, has moved from I think it's 1 meal a month to do not consume. But PCBs are lipophilic, and the lake trout actually have fat deposits throughout the meat. So there's really no getting away from it, whether you cut off the belly fat or anything. There's no getting away from it in those, and they are such a long-lived fish, and size wasn't a good structure to use for age. So they used to say I think it was anything under 24 inches you could eat, anything over you shouldn't, but now it's just a blanket consumption advisory.
Any other questions? All right, thank you.
Thank you.
All right, and moving right along here, we have evaluating the population and movement of round goby in Elk Creek, a Lake Erie tributary, which now y'all can see it. We have Carrie Salino.
Pointer. Oh, okay. Thank you. So, hi everybody, I'm Carrie Salino. I'm a Penn State research student. And this past summer, I spent my time in Elk Creek trying to evaluate the population of movement of round goby throughout there. So, if you do not know, round gobies are an invasive fish species that are now in all the Great Lakes, but also specifically Lake Erie, being introduced in 1993. They're originally from the Black and Caspian Sea, so our prediction about how they got here is predominantly through ballast water. They are generally benthic fish, so they sit on the bottom of the substrate and eat things like zooplankton, small fish, and also the eggs and fry of other fish. They also possess many characteristics that make them specifically harmful to the ecosystems around here. One such being is that they have a very fast reproduction rate. So there's about 5 females per nest, and within that they can have about 10,000 eggs. And this wouldn't be such a problem if their success rate for the fish was not high, but with a success rate of about 95%, all these fish are practically surviving. They are also very hardy in that they can withstand high ranges of salinity and temperature changes. So given this allows them to move into various different habitats without witnessing necessarily any survival adaptations that are needed. And this goes into how they consume our native species. So specifically, because they are able to move and reproduce fast, they are decimating our native species populations. This includes things like mussels, or our indigenous mussel species, including our endangered mussel species. It increases their mortality by about 10%. And they also will attack our native game species, or just those that exist in the lake, such as lake trout and walleye. There's also issues associated with birds, because birds will eat round goby, so bioaccumulation of things such as botulism. Gray herons eat about 95% of diet of goby, so if this is bioaccumulating up, they are at a very high risk. Similarly, we have put stuff in place in some areas to stop them from migrating. However, that's also preventing our native species from migrating, such as like the nace, which is already endangered. And they are just an aggressive species overall. So the hardness, fast reproduction, it is making them, their ability to push our native species out much more prominent than if they were a more minute species. So percadades, who used to be by the mouth of streams, are now pushed upstream where they are not able to survive as easily. So based on this, we have some information that we do know about gobies, which is essentially, there's a lot of them in the lake. Previous studies said about 2 billion, plus or minus 2 billion in the lake, so they're definitely in there in a high amount. And there's also not much interactions between the round gobies necessarily in the lake and in the tributaries. This is specifically based on otolith data we gathered in past years at Penn State. So generally what I wanted to take a look at was how gobies are necessarily acting in tributaries of Lake Erie. And with that, how their population size is, how they are necessarily moving in the tributary, and the population demographics of round gobies within the tributary. So these were 2 questions I tried to necessarily answer with this. How are they moving in the creek, specifically Elk Creek? And how are the population demographics of round gobies in the creek? So my study site was Elk Creek. That is just west of here, so west of the Presque Peninsula. And for this study specifically, I was looking at the lower side of Elk Creek. Elk Creek is definitely much larger than the expanse that I necessarily use, but our past sampling periods have shown that they do not go up much farther than about like 2 miles. So it did not seem to be a point to continue sampling where they were not going to be found. But it is a creek within the Lake Erie watershed. There's a cobble and gravel substrate that they do prefer because they like to build their nests inside rocks. And we used about 2.1 miles for this study specifically. So of Elk Creek, I split it up into 9 different subsections. Each section was about 60 meters wide and with about a 100-meter distance between each subsection. Some there is a bigger range of them. So over here you can see between 1 and 2 is wider than the rest of these down here. It had to simply do with accessibility or the water depth if it was safe for us to actually get samples from over there. But generally that 60-meter and 100-meter difference in between was kept throughout all of them. And as you can see, they're designated with specific colors for every site. And I will get into soon about why we had to do that. So for the actual experiment to test these predictions I want to look through, we first sampled at each of these subsections for 30 person minutes each. So by kick seining, because we saw last year that was the most effective way of actually getting round goby. And after we kicked, we would take the goby caught in the net and we temporarily put them in a bucket with water that was from the creek. And after kicking for 30 person minutes, we would measure them and we would get the sex of each goby. So to get the sex of each goby, use the urogenital papilla on them. And so in the males, it is generally pointed, is this like flesh piece that exists right below the fused pectoral. And the females, generally is more rounded. The juveniles, we put them as just a category of indistinguished, just between the two, most likely due to a lack of development. So after we had sexed them and after we have measured the fish, we would then tag them. So this was done using a visual implant elastomer tag, which is essentially a silicone-based gel or liquid that is put into the fish with a small needlepoint injector just under the skin. Each of these were of different colors, so the color was corresponding with the sections that I mentioned earlier. And since we only have 6 colors, to differentiate between the first 6 sections and the last 3, the first 6 sections were put on the right side of the dorsal fin, and then the last 3 that were duplicate colors were put on the left side of the dorsal fin. So they were each tagged, and then after they were tagged, they were placed back in the water. And this process was repeated for 10 weeks, every week throughout the entire summer from the period of June 13th to August 14th. And for the future weeks, if a fish was recaptured, we'd go through the same process again of getting the sex, the length, recording where they were first tagged, where they were now found, and then another tag would be placed behind the first tag to make a note in case they were recaptured again in the future, how that specific one was moving. So it'd be put on the body just behind this one right here. So throughout the study, we captured and tagged 1,545 round gobies. And of all these gobies that we did capture, we only recaptured 8. So what does this necessarily say about their movement in Elk Creek? Looking at the creek, there was 1 goby caught in each of the following sections. So in section 1, 4, 6, and 8, there was only 1 recaptured. And in section 2, we recaptured 4. As far as movement goes, 6 of them did not move, so they were found in the section that we had originally tagged them in. The only ones that did move was that section 6, then went to section 5, so it just went downstream a bit more, and then section 4 went 3 sections up to section 7. So what does this tell us? Fortunately, not really anything. Due to the small sample size, I'd like to be able to say that maybe it indicates since a larger population of these did not move, it would be indicative of a similar studies that show they did not move, but I cannot conclude that based on only capturing 8 of them. But based on the amount that we did catch and based on the recaptures, it generally shows a large population size. So using a Schnabel index, I was able to get an estimate, and we estimate about 133,000 goby within Elk Creek, with a lower confidence interval of about 78,000 and an upper about 433,000. I would say that it'd probably be closer to this 433,000 or 100 between the two of these numbers, just because when we were estimating this population size, I was only talking or sampling in specific subsections of the creek. So technically this number is more representative of the subsections and not the entire expanse of Elk Creek. So I assume it would either be on the upper end of this number or just closer to the higher end of this confidence interval. But we can also take a look at the variation of abundance caught throughout the summer. So specifically looking at the period of about June until August, our sample size definitely did decrease from the amount that we tagged throughout the summer. So essentially our catch per unit effort went down, with us catching about 331 goby in the first week and then only catching about 94 in the last week. So there is a general decrease in trend. Yes, there is a spike in July. I'll be honest, we can't necessarily say what is going on here with it just because there's too many outside factors, but there overall is a decline. We can also look at abundance relative to the sex of the different groups, being the juveniles, females, and males. Overall, there is no difference between them. So if you did a general capture, you're just as likely to catch a male as you would a female on a juvenile, the p-value of about 0.81. The only difference we did see was during this week 1 period where we caught the most, and that was because there was just about 182 juveniles, there's 100 females, and 49 males. But for the overall study, this is relatively insignificant. There is a difference relating to abundance regarding the section. So generally, we did see a higher abundance of goby located at the mouth of the creek. So at section 1, so at the direct mouth of the creek, we collected 255 over the course of the summer, so that gives about 26 fish collected per week. Comparatively, in section 9, there's 59 collected over the summer, so that's about 6 fish per week. And this trend continues throughout the sections, that as you move farther away from the mouth, there is less collected. There's only one exception, that's being section 3, about 62 collected, and that's about 6 per week. However, I would say that's not necessarily representative of how many goby are actually existing there, more so representative of the difficulty of just collecting them in the first place. Section 3 had much deeper water and was also just a more rocky and slippery terrain, so having us actually try to collect them was much more difficult than any of the other sections. We can also check out length variation between each of the sexes or just the groups during the summer. As generally expected, there's not much difference between the males and the females, so nothing significant, about 0.69 for a p-value. However, since they are younger, it is generally expected that the juveniles will be significantly different than adults of the males and the females, which is seen. The one thing that we did not necessarily predict based on age was that the juveniles did decrease significantly in size over the course of the summer. Again, this can only be up for speculation in terms of maybe nutrient availability or if any new groups have hatched, but generally this trend is just seen and possible outside factors could be influencing this. So what are general takeaways from this? Unfortunately, with movement, which was my main goal of this study, it is generally inconclusive. Collecting 1,500 goby and only recapturing 8 is a 0.5% recapture rate. So I unfortunately cannot tell you how they are necessarily moving, but I can at least say that based on this data, we are expecting a population size of 133,000 goby within the creek, and probably on the cautionary size of the upper end, closer to 433,000. We also saw that there's no significant size difference within the creek of males and female goby. There is, of course, between the juveniles, but that is expected since they're most likely smaller because they're not as old as the ones that are more developed. And generally also, if you were to go out and catch the fish, there's not going to be a much difference between if you caught a male versus female versus juvenile in terms of proportion in your catch group. CPUE, or catch per unit effort, did decrease from June to August. And then also the amount we did catch decreased in terms from the mouth until we went upstream. So if we wanted to say that if you wanted to catch as many goby as you could when you went out, and let's just say there are probably plenty for you to catch. Probably starting somewhere in June, higher at the mouth, so closer to the actual lake would get you a higher catch. But unfortunately, I cannot tell you if you should be moving a certain direction to go along with a higher group of the fish. So this is generally what I use for background information if anybody needs to reference it later. I also would like to take time to thank my professors, Dr. Newtow, who's back there, and Dr. Beatty, who also spent time just helping me design the experiment and process all the data. And thanks to this very big group of students that also helped me kick seine and collect the goby and tag them and measure it all through the 10 weeks of the summer, because this was definitely not a project I could have done on my own. So, any questions?
Considering your study was inclusive, what is your ideal future study?
I would be curious, one, about just running this study like again in another year, either at Elk Creek again, because I do want to see if it was just a year thing where we recaptured so little, and maybe similarly about other creeks within Erie. Elk Creek was specifically picked in that how expansive it is and that we know that there are a lot of fish there, so it wasn't like we were going to capture nothing. But also there are generally other creeks like Walnut Creek we have sampled in the past and we generally get a good turnout, so maybe something like that might be more indicative of movement overall. So if I could either try another creek or do Elk Creek again and see if the results stay the same throughout the years or if they're actually changing over the course of the year.
From the mouth of the creek until your sample or your site 9, were there any barriers that would prevent them from running through the like from 1 to 9? Log jams or physical barriers that they would be able to?
Nothing significant. Generally, like the Elk Creek, so over the course of summer, there was definitely a variation in water level. So of course, as like it was hotter out or we had some dry periods, some of the areas dried up, but there was always a connection between each section that ran through the whole creek. That one is not really interrupted by anything man-made or anything that's obstructing it naturally. Like occasionally there'll be like a tree in the water, but nothing that the goby could not necessarily work through.
All right, any more questions?
Okay, thank you very much.
Thank you.
All right, and our next presentation is the use of eDNA as a detection tool for prioritizing invasive round goby surveys in the French Creek watershed. Casey Bradshaw, what's
Sarah Lee's pointer on this?
yes, it's the middle button. It's the
Okay, perfect. All right, I'm fighting a cold, so. So first of all, there were some collaborators on this project that I want to acknowledge, and I'll mention that again. But Doug Fisher from Pennsylvania Fish and Boat Commission was a big part of this project, and then Vicki Mueller is the manager at the Erie National Wildlife Refuge, who also was instrumental in helping with this project. So we're kind of moving out of the Lake Erie Basin and moving into French Creek, which if you live in Erie County, much of the county is actually in the French Creek watershed, although most people in Erie think that they're part of the Lake Erie watershed. But if you kind of zoom in here, you can see like this is where we are, Northwest Pennsylvania. French Creek starts in New York State, and then it flows down to the Allegheny River in Franklin, Pennsylvania. And it does move through a lot of Erie County, which you can see there, Crawford County, Mercer County, and a little portion of Venango as well. So again, we're not in the Lake Erie Basin anymore. We're actually in the upper portion of the Ohio River Basin for this particular study. So round gobies, it was like a perfect way to start this presentation, having a round goby talk before mine, which kind of gives you the background of all the goby stuff. They were introduced to Lake LaBeouf, which is in the French Creek watershed, and first discovered in 2013.
by the summer of 2014, there were surveys that sort of had been instrumental in figuring out where these things were. So this is in Waterford, Pennsylvania, so not too far from Erie. They were discovered in Lake LaBeouf. Lake LaBeouf has an inflow and an outlet. The outlet is LaBeouf Creek, which flows into the main stem of French Creek. And so from this lake where round gobies were discovered, it flowed out of the lake. They're colonized quite heavily in LaBeouf Creek, and then they did reach the main stem of French Creek in 2016. So LaBeouf is like right up there in that picture, and then this is the main stem of French Creek. And that's sort of where this introduction began and where the project kind of started. So over the last decade, there's been numerous studies on the colonization of this new introduced species. I'm just focusing on a little portion of all the work that's gone into all the round goby stuff in the last decade. And one of the things that really prompted this is that when the gobies were first discovered, there was sort of a watershed-wide survey that took place through a lot of colleges and universities, through nonprofits, and also through agencies like Fish and Boat Commission and DEP. So essentially, during that time, we sampled all the major lakes and all the major tributaries as much as we could, and realized that as of then, based on our surveys, they really were still just confined to this lake and then how far they were moving down into the creek of LaBeouf. So over the years, I did a lot more surveys, lots of work with students, lots of other projects throughout the watershed. 2019 still was pretty sure that they only were in LaBeouf and working their way from LaBeouf downstream. All right, so here we have Waterford up here. The black dots mean there's a presence of round gobies. And then it's hard to see, but there's tons of sampling throughout the rest of the watershed that yielded no round gobies. However, it's very difficult to collect these fish. They are benthic, like it was mentioned before. Even if you're shocking them, they don't have a swim bladder, so they don't pop up for you to easily capture them. They kind of fall to the bottom of the stream, and then they get, stuck in rocks and logs and all of that. They're kind of a pain in the butt. And so putting intense survey efforts into these studies is incredibly time consuming, and you need multiple gear types to be able to efficiently do this. So the objective of this was to use environmental DNA, so eDNA, as a detection tool to help prioritize surveys so that we could then figure out where we want to survey to see if round gobies are actually present or absent in certain areas, so that we really can get an understanding of how far these things have moved and if they've been introduced into the other lakes or tributaries within the watershed. So methods for the environmental DNA portion of this, 60 samples were collected through from New York all the way down to the confluence of the Allegheny River. I'll show you some maps in a second. They were collected in these bottles here. They were filtered at the Erie National Wildlife Refuge. And then they were stored at -80 until they were processed at the U.S. Fish and Wildlife Service Northeast Fisheries Center in Lamar, Pennsylvania. All right, so that's towards the middle of the state. There were 2 DNA markers that were used to determine the presence of round goby DNA within these water samples, like from the filters themselves. So one was the Reese COI and the other was the Nevers. These are 2 validated markers that U.S. Fish and Wildlife Service has come up with themselves. The reason that they used 2 was to make sure that yes, both were working, but also was one more sensitive than another, all right? So down here you can see, like in July 2021, 60 samples were taken, control samples were taken, and 2 eDNA markers were used. However, the next year when we did the same number of collections, the ReCO1 marker was the only one that was used because the year prior, that one detected more round goby DNA and also validated having a little bit more sensitivity to the DNA. So there's no point in using both of them in the second year. It's a lot more expensive as well. So based on the results, and again, I'll show you maps here in just a second, the results that we got from the eDNA prompted really what my focus is of my work as a researcher, and then Fish and Boat Commission as well, is prioritizing survey efforts. Like, where in the watershed should we start looking for these, right? So to do this, you have to use multiple gear types, like I said, and this is time intensive and it takes a really long time to get this work done. So we use three different methods: an electrified benthic trawl, which we're doing here. So again, you throw the trawl, which is a big net, off the bow of the boat, go backwards. There's electricity that hits the net, and so when the fish gets electrocuted, essentially, it floats into the net. You pull the net in, you can identify everything. The other really important piece of this, that's really a completely different study, is that we identified every single fish that we got through all of these surveys, all the way to species. Which again, like, this is probably the most extensive survey that's been done on French Creek, at least within the last 50 years. So that's the electrified trawl. Also went back through the exact same spots and did boat electrofishing. So that targets a different sampling method and a different type of fish, more along the shorelines, and then fish that are sort of towards the top of the water. But we could also get into really cobbly areas with boat electrofishing versus a benthic trawl, which is more in the middle part of the channel, and you're kind of getting the bottom of the stream. In areas where we couldn't get a boat in, we use backpack electrofishing as well. So we really tried to at least get two gear types wherever we could. So these are some of the maps that were generated from the eDNA results from U.S. Fish and Wildlife Service. So on the left, these are 2021 eDNA results. The blue circles there are detections of round goby DNA. The yellow triangles mean there was not a detection. And you can see, again, up here is where the introduction was. All right, there's lots of yellow all around the other lakes, all around the other tributaries. But you can see there's detections essentially the whole way down the main stem of French Creek. This was extremely shocking to me and surprising, because I had not ever detected any round gobies in any of that water in the past decade. All right. There also was a hit here in Sugar Lake, which is like the most remote lake in the French Creek watershed. So I was really surprised by that as well. We hit that lake extremely hard after we got these results. We hit it with benthic trawl. We hit it with backpack electrofishing. We had 4 different boats out there with DEP, Fish and Boat, myself. No detections were made. We did not find any round gobies in Sugar Creek. However, there was a crap ton of geese there. So one of the things that you have to remember about DNA is if something eats something else and then poops, now you have round goby DNA. That very well could have been what happened here. The second year, 2022, same number of sites. I picked a little different locations. I hit that Sugar Creek and lake area extremely hard. No detections at all of any round gobies that second year. And again, you can see now there's some spots in the main stem of French Creek where there's not detections, right? So one year there's detections, the next year there's not detections. This is the other map that was really important to help us prioritize our survey efforts. So this is the number of replicates that was found in the DNA samples. So the darker the color, it means there was more replicates, a stronger hit, a stronger positive indication that there was round goby DNA there. So you can see up here, LaBeouf, everything was an 8, that really dark red color. Moved down through, there's some places where there was maybe only 1. So a little bit of DNA was found, not a whole lot, maybe a 5, maybe a 4. And so again, looking at the concentration or the number of replicates of DNA really helped us prioritize survey efforts as well. All right, so what did we find in all these survey efforts? So finished some more surveys this year. We have one little stretch left to do to get to the confluence of the Allegheny River. So I just have this left to do. We did find that round gobies have made it all the way to Cambridge Springs and just below Cambridge Springs. If anybody's familiar with the area, I know some people in this room are, the Venango Riffle There has been a detection there at this point, and that is the most southern occurrence that we know of so far. So they are in the main stem, they are moving. We did find different age classes, but we only found 8, right? So they're not in high concentrations at all, and they're really hard to find. Just the habitat that they like is really difficult to get into. But we did find them. So we would find, 3 within a day's survey, 2, 1, 1, 1. So there's not a whole lot, but they are present in the area. No detections at all in the lower portions of the watershed so far. We can talk about what that means here right now. So a publication is coming out in Northeastern Naturalist as we speak regarding juvenile drift as a means of range expansion in French Creek. So round gobies will utilize juvenile drift. So they lay eggs, the eggs hatch, the juveniles come up out of the substrate at night, and they just use the water as a way to get themselves downstream. Is that a reason that we have so much DNA in the mainstream of French Creek during the summers when we had collected these samples? It very likely could be that the juveniles are creating a lot of that DNA, and then they're getting eaten, right? So they're not growing into adults. There are a lot of things that will eat little tiny juvenile gobies. So to really get at this question, I did collect eDNA samples in May before hopefully breeding began. I couldn't get in there earlier than that based on how long it took Fish and Wildlife to get me a contract, and also really high water levels. So I want to sample this stuff when water levels are low, so that the DNA isn't like all diffused within the water. You want to get as high concentration as possible. We also are planning to do a spring 2025, hopefully in like February, early March, before we know breeding occurs, to see if that changes the amount of DNA in the water, specifically for the main stem of French Creek. Again, this year we did complete another 13 kilometers of sampling in the lower portions of French Creek, did not find any gobies, and then plans for next year to finish until we get to the Allegheny River, right? So conclusions to this project itself, it is concerning. So round gobies right now are in LaBeouf Lake. I really truly believe that the lake itself is what's pumping out the gobies nonstop. If we can prevent further introductions to the other lakes in the area, that is really key to keeping this invasive in check in the French Creek watershed. Prevention is always the best method, the best approach that we can do with any invasive. However, we do need to, still consider potential management strategies. So the talk that just went right before me is really interesting to me. I'm also doing a movement study of round gobies into and out of Lake LaBeouf. And so we have an array system put out in LaBeouf Creek. I have gobies that are tagged, much like you would microchip your dog. Every goby has a unique tag in it, and so when it crosses that array, it tells me what goby it is, what time it was moving. I have weight data, length data on that, and so we can see if there's seasonality going into Lake LaBeouf. If they are going back into Lake LaBeouf, that would be during a certain time of year. That would be a prime time to try to hit Lake LaBeouf and get as many gobies out as possible. So that's kind of where the management strategies are going from this point, hopefully, right? And then, it has prompted a lot of new studies on round goby work as well. And with that, I'd like to acknowledge a lot of people that have helped in the field. Certainly, Doug Fisher has been instrumental he's the non-game fisheries biologist for the state. And so he comes up from State College, and that's a lot of dedication from him and his crew. Funding from U.S. Fish and Wildlife Service and also through the Richard King Mellon Foundation. eDNA work is not cheap, and so there's been a lot of money that went into this as well. And then all these field crews have been really instrumental, especially everybody that has electrified trawls and boat electrofishing, because that gear is sometimes tough to come by, and getting everybody on board to work on one project is, I'm very grateful for that. All right, and with that, I will take any questions. Yes.
Wondering if you know what like the false negative in the DNA part.
There's so much that goes into the the DNA part of it. The false negative isn't something that comes up a whole lot, as far as I know. I did ask that question to the geneticist with U.S. Fish and Wildlife Service. I'm certainly not the expert on the genetics side of the project, but I did ask them that, and they said it's not something that comes up very often. Their methods are very precise, and however, there's a lot of ways that DNA can move around, and so that's certainly, it's not necessarily that it was like a false negative. It's that we don't know where the DNA came from, if that makes sense. Yeah.
I guess adding on to that, is your Reese CL1 marker species specific?
It
or
is.
more general?
It's species specific. Yep. Good question.
So, earlier you had a graphic comparing 2021 and 2022, like in that
Yep.
area and stuff. You said that geese might be the reason why there's detection spots in the first place.
Mm-hmm.
Were there less geese in 2022?
No, not that I necessarily recall. Yeah. There's a lot of geese. Yes.
Any observations of tube-nosed gobies? Or is it all
I have not seen any tube-nosed gobies so far. Yeah, that's a good question. Yeah, that's a good thing. Yeah. That's right.
The other Great Lakes watershed.
In Pennsylvania or
Anywhere it's becoming an issue.
New York is battling it bad in some of their inland lakes. Lake Champlain is a big one right now. So yeah, they are they are starting to be introduced elsewhere. Another big problem is that they're really good to be used as bait to catch bass. And so fishermen have figured that out now. And so there is a fear that gobies mean big bass, and gobies are a good bait to catch bass. And so, the fear is that that will start another kind of wave of spread in other places as well.
So, I guess due to how prolific they are, if this went unmanaged, what's the worst case scenario? Like, how big of a threat is this?
So French Creek is interesting. The damage in Lake Erie, it's pretty bad, right? In all the Great Lakes, it's pretty bad. In the tributaries, it's pretty bad. French Creek is unique. French Creek is the most biodiverse stream in the state of Pennsylvania and in much of the northeastern United States. It also houses more mussels than most other streams, almost 90 species of fish. So my theory is that the stream is so biodiverse that the natural biodiversity is kind of throwing elbows, and they don't let the gobies get a stronghold in French Creek the way they have in other places like Elk Creek, which has a lot lower biodiversity. And so, yes, they're in French Creek. Yes, they are impacting native species, but it's not decimating populations like we have seen in other introductions, including Europe. I mean, in Europe, they're wreaking havoc everywhere. In French Creek, their spread seems to be a little bit slower, and the densities are a lot lower than we've seen in many other introductions. And I truly believe it's because of the biodiversity.
A quick question to follow up on that. Lake Lefebvre, I know there's a lot of fishing there. Is there predators that are eating gobies?
Yeah, I think that there probably are predators that are eating gobies. Lake LeBoeuf is an interesting one. I find juveniles in the lake all summer and no adults. They're in the outflow. But I can't figure out the movement piece of them there, which is why I'm anxious to see. So that array system that I have installed right now, I'm leaving for a full year to get movement in and out of the lake. There's not a lot of good habitat for gobies in Lake LeBoeuf. There is in LeBoeuf Creek. And so it's interesting to me, like the adults seem to be in LeBoeuf Creek. We find tons of juveniles in the lake from benthic trawling. And I mean, one trawl, I can get 400 gobies, but no adults. But the adults have to be coming in at some point, I would think, to breed, or are the juvenile, I don't know. I don't know. They're a mystery to me.
All the gobies that you catch back to the water.
When I tag them, yes. Which is really hard. It's really hard to put invasive species back in the water. Right? You're just like, oh, I want you to die. But I have to put you back. But everything else I kill.
Okay.
Yes. Yep. All right. Any other questions? Yes.
How do you dispose of them? Like, is there, like...
So, like, if you found one, yeah, if I have to have permits to kill them humanely. However, it's interesting because, like, the law says don't throw them back, so you could just throw them up on shore and deal with, just kill them that way, or, yeah. Right? But I usually use them for something else, like a diet study or, I don't know, do something else with them. If I have them, I try to do something with them.
All right, thank you very much.
Yep.
I've lost my mouse. There. All right, and then for our final talk of this session, we have Angela Lair talking about predation by invasive round goby on bivalves and tributaries of the lower Great Lakes Basin.
All right. Yes, so I'm going to be talking about goby too, as if you haven't heard enough about goby already today. Before I get started, I'd like to acknowledge my collaborators. Matthew Scott is another at Buffalo who is doing fish surveys, which is where I sourced these goby from for my study, Dr. Corey Krebenhoft, and then my advisor, Dr. Isabel Portahanas. So you've heard a lot of the background on round goby already, so I won't rehash the nitty-gritty details. So they were originally introduced to the Great Lakes in 1990-ish into the St. Clair River, and by 1994 they were in like all of the 5 Great Lakes. But they haven't stopped there, as you've heard. Oh, that's interesting. As you've heard in the last couple talks, they've started a secondary invasion of the tributaries as well. And so this is Erie County, not the Erie County we're in right now, but Erie County, New York. And the red, orange symbols that you see on the map are places where round goby have been previously detected. So for my research, I'm focusing on the Tonawanda Creek watershed, which is this top stream here. It flows into the Niagara River. And then this tributary here, Ellicott Creek. And so when it comes to round goby diet, there are a lot of studies that have been done on round goby diet. Within their native range, we know that they eat up to 60% of their diet is mussels. They also eat aquatic insects, fish eggs, snails. In the Great Lakes, we know that they have a strong prey preference and eat a lot of zebra mussels. And that's part of what has facilitated their expansion through the Great Lakes, we think, as well as a bunch of other things. The Great Lake tributaries, though, many of them do not have zebra mussels. And so there's sort of this question of, in the absence of those mussels, what other bivalves they might be eating. So this leads me to my research question, which is, what is the role of bivalves in the diet of round goby in lower Great Lakes tributaries in western New York? So again, focusing specifically on the Tonawanda Creek watershed. So there are 3 types of bivalves in Great Lakes tributary streams in western New York. The sparids, so family Sparidae, fingernail clams, which are native, the unionids, the freshwater mussels, which are also native. And then in some parts, unfortunately, maybe not the parts that I've surveyed, there are invasive basket clams, Corpicula fluminea, that are also invasive, in addition to other non-bivalve mollusks like snails and limpets. And so when it comes to the Tonawanda Creek watershed, there are a lot of unionids there. Not nearly as impressive as the French Creek watershed that you just heard about, but it is still the greatest diversity of mussels in the Lower Great Lakes watershed in New York, with 21 documented freshwater mussel species. Which brings me to why we should care about what bivalves the brown goby are eating. So previous studies out of French Creek actually have shown that brown goby do consume unionids in tributary streams in the Ohio River Basin. And so of those species, there are 2 of those species that they found brown goby ate in French Creek, the elk-toe and the creeper, that are also present in the Tonawanda Creek watershed. There's also one species that was consumed by brown goby in a lab study, the plain pocketbook, and then one that's a close-ish relative of a species that was also consumed in that same study. So we have a lot of similar species. And as I previously mentioned, unionids are native. They're also very imperiled. They're important for the ecosystem. They're ecosystem engineers. They filter feed out a lot of contaminants and harmful things. They provide a lot of habitat. And they are, as I said, very, very threatened, declining in North America and worldwide. And brown goby, of course, affect them not only by eating them, but also by extirpating native fish species, which mussels need to reproduce because they have this larval stage glochidia that depends on attaching to fish gills. And so they're also hyperaccumulators. I think some of the other presenters have touched on this a bit, but because mussels accumulate, because they're filter feeders, they tend to hyperaccumulate a lot of contaminants, nutrients, even bacteria and bacterial toxins. The notorious example being some of the bird diseases that can be transmitted from mussels to goby, and then as they're starting to get incorporated in the food chain, or as there are things that are starting to eat them, up the food web. So introducing some of those new pathways. So bringing us back to my research question, my specific objectives were to determine which bivalves and potentially expanding into other mollusks that brown goby are consuming, via content analysis, and then also evaluating whether consumption of each type of bivalve is related to the in-stream density. So is there any kind of prey preference, essentially? So this is sort of the zoomed in view of my study area. So again, this top one up here, Tonawanda Creek, and the water is flowing this way into the Niagara River. So the yellow-orange diamonds, those are the previous occurrences of brown goby. These green sites are spots where we found brown goby, and then the black triangles are places where we did not find brown goby in Matt's fish surveys. And so I chose 3 sites from Tonawanda Creek that had brown goby, and 3 sites from Ellicott Creek that had brown goby for this study. Which brings me to our methods. So as I mentioned, the brown goby for this study were sourced through Matt's ongoing fish survey, which was a lot of kick seining. Basically, we'd catch the fish in the net, we'd pull the wrong goby out, we'd measure them, we'd weigh them after they had been euthanized in MS-222, and then I would remove the guts in the field, preserve them in ethanol for my later dissections, and the rest of the fish would go with Matt. We also took habitat and water quality data while we were out there, and recorded, as I said, details about the fish. There are also ongoing mussel surveys in our lab, so this will give us an idea of density, so specifically quantitative surveys with square quadrats. We also did density surveys for other types of bivalves, so I went out to the sites, and at each site extracted about a little over an inch from each of 12 quadrats, put it through a 2mm sieve, and then picked out all of the bivalves. And then we sorted them under a dissecting microscope. And then, of course, the goby guts themselves opened up under microscopes and sorted and ID'd and photographed any bivalves that I found. So these are some of the pictures that I took with my microscope. This is snail up here. You can see there's some more intact shells. These are spherids. These are fingernail clams. You can tell because of the teeth. Spherids will have little like arc-shaped teeth, whereas the invasive Corbicula will have more triangle-shaped teeth. There's also the lateral teeth, the number of them. Unionids will only have lateral teeth on one side, so they're pretty easy to spot as well. But a lot of what you pull out also looks like this, which you can't really identify, but you can say, you know, these are some shell fragments. This is some kind of bivalve. So these results are very, very preliminary because my gut analyses and ID of specimens coming out of the bivalve surveys are still ongoing. But these are just kind of some first pass figures. So for each of these, we have on the y-axis the number of whatever organism it is found in the guts per number of goby. So like on average, for example, this one is just total bivalves. So for example, in the first Ellicott Creek site, there were like about a quarter of a spherid per goby that we found, for example. And so we have the data here for snails, for the fragments specifically, and the spherids, which combined make up this bivalve data set. So you can see this one here is kind of an outlier. That's because we only caught one fish. The ends here, these are sample sizes. So this one we caught one fish and we found one spherid in it. So that one's a little out of the ordinary, but the others we caught, 6, 7, 8 goby, 9 goby. So these are the preliminary results of the bivalve survey. And so the blue sites, the sites shown in blue are the 2 sites in Tonawanda Creek. The 3 sites in gray are the Ellicott Creek sites. And you can see that all of the non-unionid bivalves so far that we found in the goby guts have been spherids. And also in the quadrats as well. We haven't found any Corbicula yet, any of the invasive clam. But you can also see something kind of interesting about this, which is that there are some crazy outliers, right? Like most of the data is pretty clustered, pretty low, and then you have these crazy outliers. And so what this tells us is that when we are finding spherids in the environment in these quadrats, they're being found in really large clusters. So you either find 0 in a quadrat or you find a ton of them in a quadrat. And then here is just my very rudimentary attempt at drawing a relationship between some of these things. So I was curious to see if with increasing bivalve density, if sites that had a higher incidence of bivalves also had, if goby from those sites also had more bivalves in their guts. And as it turns out, there's a non-significant but negative relationship. So this one, your y-axis here is goby consumption for both of these. Just the proportion of gobies eating bivalves is like 100%, 50%. And then this is like increasing bivalve density. And this is the average for the site. And then this is the maximum. So the most we found in a quadrat per site. Again, the reason I split it up this way is because there was such a differential, some of those really huge outliers. And I wanted to see what it looked like on a scatter plot. And so this result was kind of surprising to me because you would expect, right? And this is, you know, like I said, still very early phases of data analysis here, but this was really surprising to me that we were seeing some of the sites with the highest densities of bivalves in the environment had actually some of the lowest incidences of being eaten by goby, and then sort of vice versa. So some of the sites that had fewer bivalves in the quadrats actually had more bivalves in the goby guts. And so what does this tell us? One theory would be that there might be other prey that are also abundant at sites wherever the bivalves are abundant. So maybe the bivalves are abundant at sites where lots and lots of things are abundant. And so, you know, bivalves, they're not exactly easy to like chew up. And so maybe they get more bang for their buck by eating, so to speak, by eating, you know, chironomids and macroinvertebrates, non-bivalve macroinvertebrates, things like that. And so that's one possible explanation, but again, very, very early data here. And so in terms of into the future, so I need to finish my data collection. And because I've only found spheroids so far, I'll probably expand the study to include snails as well so that I can compare sort of, you know, ask some prey preference questions as I originally planned to do. It would just be probably spheroids and snails, assuming I still have a few more guts to go through. I haven't totally given up hope, but assuming that I might not find any unionids or any invasive clams, it raises questions about if round goby are not consuming goby, or round goby are not consuming unionids in Tonawanda Creek, why that might be. So this will be informed by the mussel surveys that are ongoing in other parts of the lab. So I can investigate other factors, like are the individuals that we're finding just too large, because mussels do grow really quickly and they grow really big. Maybe there are larger species in general. A lot of the really endangered species found in Presque Creek are also like kind of itty bitty, and we don't necessarily have those here. So maybe, you know, they're growing too big, too fast. Maybe there isn't enough recruitment, but these are some of the things that with Max's data I can maybe answer, or try to answer or speculate about. And then also investigating effects of individual or site characteristics. So we collected all this other data about the mass of the fish, the length of the fish, the sex of the fish, all these like abiotic and habitat water quality parameters. Do any of those things influence how often goby are consuming bivalves? Do males or females consume goby or bivalves more often? Some studies have shown like mass and length might have an effect, so it'll be interesting to see if we see that here as well. And then finally, other questions for the future. If I don't have any sites, unfortunately, with the basket clam, that still raises the question: do they eat the basket clams? And how does this affect the dynamics of sites where you have zebra mussels that are absent? Interactions between the invasive basket clam, which competes with the unionids, but then the round goby also impacting the neonids, and then but also potentially impacting the basket clam. So you might get some more complex interactions there, and I'm curious about that in the future, but that's way down the road. All right. So with that, acknowledgements to my a lot of my people who assisted me in the field. My undergraduate assistant, Trey, Fen, the entire fish and mussel teams, with Matt and Max and Jonah kind of leading the way, and then my wonderful lab assistant, Chanelin. And so with that, do you have any questions? Oh, I went too far. This is like my extra data. Okay, hello. There it is, okay.
Do you think that the presence of zebra mussels where there are not known yet are predicted for?
That's an interesting question. I mean, I think wherever you have zebra mussels where the round goby then get introduced, I think they'll have a plentiful food source then, right? So they might, sort of like how they did in the Great Lakes, right? They might go crazy. But that's not really something we would probably see in most of the tributaries, just because a lot of the tributaries don't really have the zebra mussels. Some of them do. And in those tributaries, if the round goby were to be there too, they might spread faster, but there are other, as you heard in the other talks, there are like a lot of complexities to the way that they move throughout the watershed and those dynamics as well. So that makes it a little different than in a lake. So it's a really good question. I'm honestly not sure. Yeah.
I'm not familiar with those two streams, so like how deep are they, or what's the substrate? Is it real rocky?
It depends on the site as far as the substrate goes, and that's why I really, really want to look at some of these like other factors that might influence things. Substrate type, I think, is going to be a big one, just because I think it's really going to influence where you find what bivalves, where you find bivalves at all. Also where you find goby, right? Because goby have preferences for a certain type of habitat as well. So yeah, I think, sorry, I forgot the second part of your question.
Yeah, I was just, what the substrate, if it was rocky or muddy, or if it was like a constant flow, or if it was
Kind of a mix. Varied depths as well. Part of the reason we didn't survey any sites where the basket clam has been reported in the Tonawanda Creek watershed is because those are all like the really deep parts near the mouth into the Niagara River, up in the Erie Canal, which you can't really see on the map, but extends off of Tonawanda Creek to the north. So.
Any other questions? Okay, thank you very much.
All right, so that does finish up this session for us today. We still do have one more session for today. We are going to break before then, and I do want to note that the first talk of the next session informed us that they are unable to make it. So I'm going to extend our break a little bit. So our next session will start at 1:55. So that's a little different than your proceedings. And there are refreshments in the back, so please help yourselves. All right, thank you.
We're gonna start our last session for our 20th annual research symposium. Let me get to the right page here. I do want to remind everyone that after this session at 3 will be the presentation of the Student Research Awards. So if everybody wants to stick around for that, I'm sure we'll have some more people filtering in as well. But without further ado, we'll get started with this last session. Max Streidel will be presenting characterizing one of Western New York's most diverse mussel assemblages. And this picture here, there's a laser pointer, and you can advance forward and backwards with the side buttons.
Thank you.
Yep.
All right. Hello, everyone. My name is Max Streidel. I'm a master's student at the University of Buffalo, and today I'll be talking to you about my work characterizing one of Western New York's most diverse mussel assemblages. So to get us started, I wanted to give everyone a quick introduction into unionid mussels. So mussels in the family Unionidae are highly diverse with around 300 North American species. They provide a variety of ecosystem services, such as cleaning water through their filter feeding. They provide habitat for other invertebrates, and then they also are involved in nutrient cycling. But unfortunately, they're also highly imperiled, with 96 species federally listed as either threatened or endangered, as well as a multitude of extinctions that have already occurred and many more non-listed species at risk. They also have a really interesting life history strategy that the people at Vox describe as strange and savage. And while I might not use those words, it is a very interesting life history strategy in which their larvae or Glochidia undergo a parasitic life stage in which they attach to a host for dispersal and nutrients. This host is usually a fish. So on the right there, you see some of these larvae, those little specks attached to a fish gill. And then on the left, you can see this is a picture of a fat mucket mussel, partially buried in the sediment. And a lot of times the females will extend what's called a lure to attract a host so that their larvae can attach to that fish. So some of the threats to unionid mussels, one of the most important is habitat degradation, channelization and impoundment, pollution, sedimentation, and the loss of riparian zone can all cause mussel declines. And then invasive species such as zebra mussels and basket clams can also cause declines through competition for food. That picture there is a mussel we found over the summer, and what's circled there is where you can see where a zebra mussel was physically attached to that mussel. And then I wanted to include this quote at the bottom, which basically outlines the state we're at in mussel conservation currently, where we still have tremendous diversity, but this diversity is very much at risk, and we could be losing, if we're not careful, half of this fauna in the next, well, it would now be 20 or so years. Another phenomenon that can occur in mussel declines is you may still have adult populations that are present, but these populations may not be actively recruiting or reproducing. So this is an example from southeastern New York, and you can see on the right there that the youngest individuals they found were 30 years old, which means that this population is not sustaining itself. A healthier population would be this bed on the left there, which has more of a good range of young individuals. Some of the causes that have been floated for potential reasons for this phenomenon are a lack of host fish and poor interstitial water flow. So I wanted to give an introduction into the area in which I've done my research. The Tonawanda Creek Basin, which is the Tonawanda Creek is a tributary of the Niagara River. It has significant regional diversity and also a set of surveys done in the past that we can compare our data to. So in 1998, there were 36 sites sampled, and they found 16 live species. In 2016, 4 of these sites were returned to. They found 17 species. And then we returned to six sites and found 16 species. And one additional note before we move on is that the majority of the surveys that were done before us are qualitative, which means you start from downstream end, work your way upstream, and are either visually or looking for mussels or just feeling the surface. And this method has been found to predominantly find larger species and larger individuals of those species, which means that we could be missing some of the diversity of these beds. And then, even though I will be comparing to historic states of 1998 and 2016, these are already conditions that have experienced a serious decline. So I wanted to put these quotes up that were both from the 19th century, describing mussel communities that are in essentially every river in the eastern United States, and that are occasionally so dense that you can walk for a mile in a river without stepping on a live mussel. So for my study goals, the first would be to update these previous surveys to continue building on this record of mussel community. And we also want to determine if these mussel beds are undergoing active recruitment. And then we wanted to see if these mussel densities and recruitment are tied to any specific habitat conditions. And then we also wanted to assess the efficacy of our methods to guide future work in this region. So for our study sites, we are basing the majority of our study sites off of these previous surveys. You can see they're pretty wide-ranging across this basin. The majority of them are in Tonawanda Creek and Ellicott Creek, which Tonawanda Creek flows into the Niagara River, like I said earlier, and Ellicott Creek is a tributary of Tonawanda Creek. So we focused on a subset of these surveys, the ones that have the largest mussel beds, so that we can search for juvenile mussels at these sites. So we have 3 sites in Tonawanda Creek, 2 sites in Ellicott Creek, and then that one site that's in the smaller river is in Ransom Creek, another tributary of Tonawanda Creek. So for our methods, we first performed mussel surveys at these sites, and our mussel surveys occurred in essentially 3 phases. The first being an initial qualitative, in which we performed a quick search of habitat to locate mussel beds and to ensure that no federally listed species were present. And then our second phase was quantitative, which is we have quarter-meter squared quadrats that we excavate to a depth of about 10 to 15 centimeters and then put this substrate through sieves. And this allows us to find very small mussels to make sure that these beds are reproducing. And then for the final step of our mussel survey, we performed another qualitative survey. And these are just time surveys from downstream to upstream. And when we perform these, because this is what the previous surveys did, and we wanted to be able to compare our data to their data more easily. We also performed habitat assessments at these sites. So that includes a variety of surface water analysis, substrate analysis, and something that I was excited to add was interstitial water analysis. So that's the pore water of the sediment. And that's something that we're still working on getting data back for. So I wanted to get into some of our results and start with a brief summary of our mussel survey. The densities from our quantitative survey range from 0.4 mussels per square meter to over 16 mussels per square meter. And then catch per unit effort, which is a measure of how many mussels you're finding during qualitative surveys. We found 22 mussels, a range from 22 mussels per person hour to 266 mussels per person hour. And then we found something that's exciting is we found 2 species that had not been recorded at these sites previously. The first being the slipper shell mussel, which we found at our Ellicott upstream site, and the second being the eastern pond mussel, which we found at Ellicott downstream. So now I want to compare our data to the 2 sets of historic surveys. So you see in the blue bars are our surveys, and then going back in time from there. And this is just catch per unit effort. So again, how many mussels we're finding per the survey time. And at first glance, this looks really concerning. You're seeing catch per unit effort decrease heavily in the majority of these sites. But there's something more going on here. So I wanted to break it up into the 2 variables. So mussel abundance is on the top and survey time is on the bottom here. And you'll see that in most cases, we were finding higher mussel abundances It's just taking us a longer period of time. And so the reason this might not actually be as negative as we think it is because the longer you spend at a site, the more muscle poor sites you're going the more less ideal habitat you're going to survey, the more low density patches you're going to encounter. Doesn't necessarily mean that there's a decline at that site. But there are 2 sites here in which we found lower mussel abundances in the longer survey time, which could certainly be indicative of a decline. And then we have additional cause for concern with species richness, or the number of species we're locating at these sites. You can see it's holding stable in our tributaries, Ellicott and Ransom, but decreasing in each of our Tonawanda sites from the 2016 surveys. This is a table that has some summary of our quantitative surveys and also of our habitat assessments. So you can see our densities are much higher in the Tonawanda sites compared to the tributary sites. But something that is encouraging is that we're finding very small mussels under 20 millimeters in all but one of our sites. One of the interesting water quality results is that the 2 sites with highest water hardness, Ransom Creek and Ellicott downstream, had the lowest mussel densities. So that could be indicative of some sort of contamination, whether it's through metals or just too much minerals in the water. And then another trend we noticed is that, again, the 2 sites with lowest mussel densities also had the smallest delta 50, which is a measure of substrate size. And they also had the highest percent fine sediment. So that could be indicating that fine sediment is a barrier to some of these populations. And then the bottom row shows interstitial dissolved oxygen, which there are no trends that we found initially from that, but that's something we'll try to piece apart more as we go on in our data analysis. So now I want to compare between our methods, between the quantitative and qualitative surveys. So you'll see in yellow, these are the sizes of a species that we found in our qualitative survey. And in the blue, we have the sizes of our species that we found in the quantitative survey. And so this is, I've broken down our species by tribe just to make the data a little bit more easy to see. You see that we're tending to find smaller mussels in quantitative, in that quadrant excavation, than we are through timed searches and qualitative surveys. And so here we have 2 significant results with Fat Mucket and Plain Pocketbook having significantly smaller mussels found in quantitative surveys. And then for these tribes here, for Spike, for Three Ridge, and for Wabash Pigtail, we are also finding significant differences between the 2 survey types. And this is the same analysis, just shown a little differently because we found smaller sample sizes of these species. So we do see that trend continue for most of these species, with significant result for the giant floater. Now I want to compare our methods and what they found for community composition. So this first bar graph here is for our tributary sites, for our 2 Ellicott sites, and for our Ransom site. On the right of each of these panels, so here, here, and here are our quantitative community compositions. And then to the left of those is the qualitative data. And so we found pretty low sample sizes for quantitative. These sites tended to have low mussel densities. But in general, we are seeing a little bit more evenness in our quantitative data compared to our qualitative data, with an exception being Ransom Creek, which had pretty much the same even with a small sample size in the quantitative. Another thing to note is that we were locating more species in our qualitative surveys, which makes sense. You're covering a larger area, and so you're going to be able to just find more species that way. And so this is the same data for the Tonawanda sites, and you can see we're finding a lot more mussels in the quantitative survey here because of that higher density, and also a lot more species. But for each of these sites, the one trend I want to point out is that green bar that's taking up most of this figure is the three ridge muscle. And in each case, we're finding a higher proportion of three ridge in our qualitative data than in our quantitative data. That's because three ridge can grow to be quite large. And in the case of the Tonawanda midstream site, we're actually seeing a different dominant species based on our survey method. So in the qualitative, we're finding a mussel bed that is majority three ridge, whereas in our quantitative, we're finding a mussel bed that is majority spike, which is a mussel that does not grow as large as the three ridge does. So in conclusion, we may have some sites in decline with Ransom Creek and Tonawanda midstream. We found fewer mussels in a longer search time, and then we did not find recent recruitment or mussels under 20 millimeters at the Ellicott downstream site. And then for species in decline, we did not detect live elk toe or rainbow mussel, and we only found a single live deer toe, which was only detected during the initial qualitative phase of one of our surveys. So some key takeaways. The most important one is that we did find mussel recruitment. You can see on the right there, we found some very small mussels. If you ignore my pruny fingers, you can actually see them. So that means that the conditions at these sites are suitable for the recruitment of the most common species. The two at the top are juvenile fat muckets. This one here is a juvenile three ridge, and this is a juvenile spike. But unfortunately, though, we may be having some reductions in species richness and abundance at these sites, which could be indicative of a problem. And then as far as the methods go, a combination of quantitative and qualitative surveys will be needed to continue tracking changes in this region. And we do have a lot more data to come with associations between mussel species and substrate type. We'll have some interstitial water chemistry coming soon as well with some ammonia, and also fish assemblage analysis from one of my colleagues who will be lending us that data. So stay tuned if this is something you found interesting. And then I wanted to quickly acknowledge my advisors, Dr. Corey Krabenhoff, Dr. Isa Portahanes, our lab technician, Jonah Frank, who've all been super helpful on this project, and all of my lab mates who've helped with this. And with that, I'll take questions. And my contact information is up there if anyone would like to talk more about this.
One of the tables that you had up was mussels under 20 millimeters. Would that all be juveniles, or are there some species that just stay that small?
There are some species that are that tend to be closer to that size, even as adults. Those are not species that we are located during these surveys. So some species like lilliput is in this region, but we didn't find it during the survey. So all the species that we found of that size would probably be a year or two.
Are any of the species hard to identify, look really similar? And if so, what's your method for that? Is it field guides, or do you have people who are really well versed in identification?
Yeah, there are definitely some confusing species that we find. And what's great is, we can, a lot of times we just, we can take pictures, we go based off shell morphology. Mussels they have beak sculptures. So on this part of their shell, a lot of times they'll have a little pattern on the top that you can use to identify them. But that being said, it does get tricky. So, you take pictures and you show it to a lot of people. And me and Jonah went to a mussel identification workshop to improve our skills too. So, but yeah, there are definitely some tricky ones for sure.
You had a lot of different mussel species listed on one graph. Are some noted as pollution tolerant or not pollution tolerant, where it seemed like there are certain species, there was a lot of them? Are they just tolerant of all kinds of conditions, while maybe other ones are kind of not so tolerant and you don't see those?
Yeah, absolutely. So a couple of the species that are more tolerant are the giant floater is known to be tolerant of a lot of different conditions. Same with fat mucket can be pretty tolerant and hardy. I'm not so certain about how hardy three ridge is, but it's certainly well suited to these sites at the very least. And then, we have had some extirpations. There are some species that we only find shells of, so I would assume that some of those are the more sensitive ones. But yeah, a lot of what needs to be done is what specific habitat and conditions are suitable for individual mussel species, which is not super well known.
Any other questions? All right, thank you, Matt.
Thank you.
All right, and for our last presentation of our 20th annual research symposium, we have Jonah Frank presenting on Are We Mussel Ready? Evaluating habitat water quality in the upper Niagara River for native mussels.
Thank you. All righty, you've all been waiting for this moment. So it has been lovely being at this conference. It's been amazing to see all the brilliant, hardworking people studying all sorts of things, whether it be meteorological conditions, bacterial assemblages on mushrooms, or addressing engineering solutions for common problems. And I've been so impressed just at the brilliant minds, hardworking folks throughout this area. And I would like to frame this talk in that same vein, highlighting brilliant minds that have worked and built up a history of science that we are benefiting from. So as we talked about earlier, we'll be discussing the feasibility of reintroducing mussels to the Niagara River. I'm a technician on a team that consists of Dr. Isabel Portojanes, Dr. Corey Krabenhoft up at University of Buffalo, and our wonderful master's student, Max Streetel, who you just heard talk. And I'd like to start off with some context, specifically context that is both historical and region specific. In about the turn of the century, Elizabeth Letson returned from eastern Pennsylvania after having visited the Philadelphia Museum, impressed by how they ran their museum and divided up their responsibilities into various taxonomic focused sections. She was inspired to start a conchological section in Buffalo, which is the study of shells, and undertook this mammoth task of documenting all the mollusks of the Niagara region and beyond. And they did a really good job of collecting things. So we have these nice records with these specimens in the Buffalo Museum of Science that were collected in the 1800s, early 1900s that we can reference. And this group of people were amateurs, there were academics. Elizabeth Letson herself was not she didn't have a doctorate. She was awarded an honorary doctorate later for her contributions to science in the region, but she herself was not a university staff, anything like that. And they had a lot of buy-in, and they went far afield. This is Clifford Blakesley sampling some mollusks in pickerelweed, and if you look up here, this is the card for this 3 ridge that you found. This was collected on Presque, right out here. So this is, if we go to the Buffalo Museum of Science, a lot of the shells that we have in our collection are collected right here, which is really amazing. Just recently, we were talking with Sarah about the ongoing mussel efforts here. This species is no longer found here. So it provides an interesting record, not only of what species are present, but how things have changed over time. And we can see that illustrated in some beautiful ways. This is another member of the Conchological Society, Imogene Robertson. Here she is looking at mollusks. There's the falls right there, not the sort of outfit I wear when I'm doing collecting, but it was a different time. And during this time, they were able to identify all these different sites and record what species were present. And there was this one site in Tonawanda Creek outside of Pembroke, New York, where she said they found some of the largest specimens she'd ever seen of fluted shell and fat mucket. However, after it was visited by pearl hunters, she recorded this. The pillage was deplorable. As the finest forms of these mollusks were nearly depleted of their patriarchal forms, a distinct loss to the conchologists of Western New York. On this small scale, she witnessed a depletion of a population, and it's indicative of a much larger loss occurring throughout the region. As industrialization took over the Niagara region, we saw pollution occur, habitat loss, redirection of water, all of which contributed to massive changes in the Niagara River and the Great Lakes as a whole, resulting in degradation of the region, which led to such a great change that there was a binational agreement made to make a change. This occurred in 1972. It was an election year, by the way. So that was Nixon. Nixon here, he was running, he actually won. And he's shaking hands with Trudeau. This is them signing the Great Lakes water quality agreement, in which they agreed to improve the Great Lakes water conditions. In 1987, they made it a requirement that each place had to come up with a plan on how they would actually improve their water quality. And then in 1994, they said, okay, we're serious about it now, let's make these plans. So in 1994, a remedial plan was made for the Niagara River. So that was exactly 30 years ago. That was done in, I can't remember what time of year it was. Anyway, we all celebrated. It was great. 30 years have passed. A bunch of different people have worked on improving this river and changing these different impairments. These are things that have been identified in 1994 that are measurable ways we can see degradation or habitat loss in the Niagara River. So this is from the original document in 1994 describing some things that need to be addressed in the Niagara River. So over time, some of these have been improved, some of them have been removed from this list. So we're seeing improvements. A lot of people have put in a ton of work, and it's amazing. So the question is, based on this, well, before we get to that question, let's talk about where our mollusks are at. So in 1948, when Imogen Robertson and Clifford Blakesley published a bulletin of what species were present in the Niagara River, there were approximately 30 species identified. In our most recent surveys from some different groups who surveyed the region, there are about 13, so fewer than 20. Probably more than half have been extirpated from the Niagara River. Some things have, and in general, the beds are smaller, which is not great. And additionally, different from when the Great Lakes Water Quality Agreement was originally made, or immediately preceding it, zebra mussels have become a much larger problem than they were originally. So now let's get to the question. Given the improvements, can we bring mussels back to the Niagara River in higher numbers? And that is what we're trying to answer. So how are we going to go about doing it? First, we're going to need juvenile mussels. We propagated these in a lab. We're then going to distribute these at sites in the Niagara River and at sites in the tributaries, which have mussels in higher abundance. We're going to measure the growth of these juveniles over time, compare mortality rates between sites, and determine what factors may play into growth and survival at these different sites. And maybe this will give us some answer on whether it would be feasible for mussels to survive in key spots in the Niagara River. So let's talk about propagation. This is a view of larval mussels. This is what they look like when they come out of their mother. They're just a shell. They have some muscle there. They don't have a foot. They don't have a digestive tract at this point. They're just open. They're made to clamp down onto a fish. Max has talked about their life cycle so far, but I think this is quite beautiful. And they produce, depending on the species, tens of thousands to hundreds of thousands. Some species produce millions. So quite beautiful, but these are fat muckets, which you'll hear quite a lot about as we go. Before we can get those babies, though, we needed to collect these. So we went to a local creek, Ellicott Creek, and we found 3 gravid females in the late summer of 2023. These were brought into our lab, and we will maintain them at about 10 degrees Celsius so we can collect the juveniles or the larvae in the spring. And because we are sentimental folk, we named these Elizabeth, Imogene, and Nellie after Conchological Society members from just as a nod to our history of mollusk science. So spring comes around, it's time to collect the juveniles. The female mussel broods her young in specialized gills called marsupia, and these larvae can be collected from the gills. You infiltrate the gills with a needle, you flush them with water, and the gills rupture and out come the babies. That's how they're supposed to work. They're meant to be ruptured by fish, so this doesn't harm the mussel. She'll recover and be just fine. We put these Glochidia inside a aerated bath, added fish to it, left the fish in for a certain amount of time, giving them a chance to be exposed to those Glochidia. Then we introduced these baths into a system where the juveniles could undergo a metamorphosis on the fish. That takes about 2 weeks. They drop off, then we had a filtration system that recovered them. Additionally, we sent Glochidia from a fourth female we collected to a site in, a facility in Missouri, a USGS facility where they have a lot of experience propagating mussels. After those 2 weeks finished, we collected our mussels. We kept our mussels inside these things called Barnhart buckets. They're screen-covered cups that sit in a bucket. There's a current running through, so water runs down through these screen-covered chambers. And unfortunately, we experienced a lot of mortality, nearly 100% mortality. So that method didn't work too well for us. Meanwhile, at the USGS facility, they had amazing success. So that is where we sourced most of our juveniles. They use this pulse flow-through system in which the juveniles are housed in beakers and they're actually allowed to live inside sediment, and a regular water flow goes through here. So this is a view of these juveniles. Once they come off the fish, they're very small. They haven't grown much from actually from being on the fish, but they have a foot, they have gills, they're capable of filter feeding, so they're ready to start off as a free-living mussel. Here's a picture of some more developed mussels from the USGS facility. So now that we have our mussels of an appropriate size, we could introduce them into the field. So we had these tools that we use for this purpose. We have mussel silos, which were developed by Barnhart, same guy who developed the Barnhart bucket. It's a cement dome with a PVC core, which allows us to put a filter-covered a screen-covered cup inside that sleeve in the middle. You set this on the bed of the river, like so, and this allows water to get pulled up through the chamber via the Bernoulli effect, and that will feed the mussels. And we have these Viber Whitlock boxes, which can vary in the sediment. The purpose of this was to see whether being exposed to the sediment changed how the juveniles survived, whether being only exposed to the water affected the survival and growth of the juveniles. So we were able to utilize these different tools to compare those effects. These were deployed from July to October of this year. We selected sites in the Niagara River, 2 in Buckhorn State Park. Am I looking at this funny? Nope. Okay. So we had a couple in Buckhorn State Park. We had some down by Beaver Island. We had one on Strawberry Island, which is only accessible by boat. That was fun. And then we were going to have one at Unity Island, but we didn't. So these are our control sites in Ellicott Creek and Tonawanda Creek. We had 5 silos, 5 cages at each site, 20 juveniles in each cage or silos, so about 1,200 juveniles were deployed. And these are our results. So pretty drastic. Ellicott Creek, Tonawanda Creek, we saw dramatic growth compared to our test sites. We don't have all of our data in for these test sites. It takes a long time to measure each of these juveniles. We're trying to be really careful to get everything accurate because there can be very minute changes in size over time. But by far, Tonawanda Creek had the highest growth rate. You can see here where they basically doubled in size over time. And this is only at day 30. This trend continued well into the end of the study. But you can see it's drastically different. And also at sites where they're outside of Tonawanda Creek, the growth rate between silos, which are exposed only to the water, and to the sediment cages, which are buried, this growth rate was vastly different. So just on this scale, we can compare this. This is juveniles at Tonawanda Creek. These are juveniles at Beaver Island. Comparatively, very little growth compared to the Tonawanda juveniles, where they go from about the same size, between 5 and 10 millimeters, while at Tonawanda Creek by a month, they're over 20 millimeters. These ones are just slightly higher than where they started. So clearly there's a difference in growth between these sites. What about survival? That's also pretty drastic. Oh, pardon me. This is what it looked like at the end of the study. This one's from Tonawanda Creek. These are ones that were in the Niagara River. Very drastic. Remember, these are siblings, okay? So they came from the same mother. Very, very clear distinctions in growth rate. Survival, we saw similarly distinct trends where we saw much higher survival in our control sites and much higher survival in silos compared to cages. Cages in Niagara River were especially hard hit. A lot of the losses were not divided. It wasn't a diffuse loss. You would have a cage entirely die. It wouldn't be evenly spread between cages, so there might be something indicative there. Abiotic factors may play some role in this. We're still evaluating what might play into that, but we especially looked at sediment quality. One thing is we did a grabbed sample. We just walked around, took random samples of gravel and measured their size, and we saw that while the size of the sediment didn't seem to affect mortality in silos much, it may have a very strong effect on survival in cages, and this makes sense. If something is buried in gravel with lots of space for water to go through compared to something that's buried in very fine sediment where there's very little interstitial water movement, that could play a factor in survival of the juveniles. We even looked at DO in the interstitial water space at these sites, and it's basically the same with some exceptions, where at the sites where there's a lot of fine sediment, there tends to be relatively low DO in the interstitial space as well. So very little dissolved oxygen in those spaces. So burial in fine sediment seems to have some effect on survival and growth as well. While juveniles in the Niagara River can survive, they might not grow as much as they do at some of our control sites, which is fine. If they survive, that's great. But they need access to the water column. They can't be completely buried. Another thing outside of our results is siltation posed a major challenge to some of our tool use. Our silos would get buried at sites, and that may have played into mortality in some silos later, not because the silos themselves were having a poor effect, but because they were buried. So it was basically like they were in a cage. So we are at a point where we're trying to evaluate what we can do next to improve our study further. Here's some more key features. Just as a comparison, I believe this is the same day or same week. These are juveniles from Buckhorn. Some of them are dead. Those are the ones that are open. Here are some that are from Tonawanda Creek. Once again, drastic change in both mortality. There was 0 mortality at Tonawanda Creek, relatively high mortality and low growth at the Niagara sites. This year, we're trying to propagate more species, plain pocketbook. We're evaluating on-site lakes for possibly grow-out ponds for our juveniles later on. And we are modifying our propagation methods, hopefully to get a higher yield. This is a breakdown of some things we might want to improve to increase that yield. And we're pretty optimistic that we'll get a higher return of juveniles this year, or rather I should say next year when we propagate the brood stock we've collected. Finally, I'd like to thank all these people, tying it back to the rich history and current status of science in this region. We have been helped so much by people here in Pennsylvania. The Pennsylvania Fish and Boat Commission has lent us valuable assistance with advice. Scott Ray and Joshua Arnold, sorry Josh, have given great advice on propagating mussels. James Coons at the USGS facility in Missouri helped so much with supplying the majority of the mussels we used in our study. We had amazing undergraduate students who helped with field work throughout the summer, counting mussels, helping to build silos. And additionally, we have wonderful students in our lab who, even though they weren't actually helping with this project, were always willing to lend a hand in a pinch. So with that, do we have any questions? And sources.
I have a question.
Yeah.
Out of curiosity, is the Conchological Society of Buffalo still active?
That is a really good question. I don't think it's the same. So it was the conchological section, as in a specific division of the science, what are they called? The Buffalo Natural Science Society. So I think, so the Buffalo Natural Science Society is still ongoing. It's what runs the Buffalo Museum of Science. But I don't think they're divided into a conchological section. Great question. Yes.
What are the predators when they're still kind of very small in that juvenile phase, and is that impacting your study at all?
Okay, great question. So let's go back. Just so we can have some visual aids. When they're this size, flatworms will eat it. If a flatworm gets into a system, it can eat juveniles like crazy, so you have to be really careful. However, we don't deploy them at that size. By the time they're going out, they're 3 millimeters long. They're placed inside basically the screen is about window screen sized. It's at that size that it keeps things from getting in that could eat them at that size. So I don't think predation has played an effect in the mortality of the juveniles. They're pretty well protected inside their the chambers or cages. Great question. Yes.
So I know we previously discussed how you can tell with some species the difference between males and females.
Yes.
How can you tell if a female is rabid?
That's a lovely question. So just with so they have those modified gills in which they store their juveniles. If you peek inside a mussel, which is hard to do, and anyway, you can look in and you can see their gills. If they have juveniles inside there, they're nice, they're puffy, they often have a black edge. And not all different mussels keep their juveniles in for different periods of time. Fat muckets generally overwinter with juveniles in their gills. They're called long-term breeders, but other species might not keep their juveniles quite that long, so they might look a little different. But for the species we look at, you can tell because they have beautifully puffy, black-lined gills. Yeah. The trick is getting them open. Any other questions? Yes.
There was a huge difference in that 30 days where the one site had very small ones and then it was considerably larger for the other site.
Are you referencing this graph?
Yeah, I think there was a picture that you were holding them in your hand and it's 30 days.
Well, not my hand, but yes, I hand holding. I'm not married. So let's see. Yes, very drastic.
Do you think that is impacting, like, big picture, the survival of mussels overall? Is like the impacts that cause them to grow slower is making them more vulnerable to?
It could be. When they're at that small size, they're more vulnerable to things like round goby, right? They're just a round goby snack at that point, or a drum. There's plenty of things that would eat a mollusk at that size. Quite possibly. It depends on the life history strategy of the species. Some species, their main purpose is they have really rapid growth early on, and then that way they get out of that window of being really susceptible to predation, but generally those are short-lived species. And the species we picked are kind of in that category, so they're pretty good so if they're growing, it's going to be pretty drastic. You are correct in saying that if they stay smaller for longer, could play a factor in predation or loss. Great comment. Any other thoughts or questions? Yeah, Angela.
I was just going to ask, why did you choose that?
Why did we choose fat mucket? Fat mucket are supposedly easy to propagate. We didn't have as much luck as we would have liked, but that's the main reason we picked it. Another reason is they are fairly tolerant of conditions. If we were to pick something more sensitive, they might all just die anyway. But we chose, if we can test and see if the most robust, most adaptive, most resilient species can survive in the Niagara, that's a great starting point. You wouldn't want to jump in with like the most sensitive species as a test. Excellent question.
Any other questions?
All right, thank you.
So that will conclude our last session for Symposium this year. We're going to be taking like a 20-minute break, round up some of our judges' scores and everything, and then we will be presenting the student awards at 3 o'clock. There is still more snacks in the back, drinks. Eat as many as you want, eat as many mints as you want. And if you have any questions for our presenters, they should be sticking around. So feel free to take that time to do that as well.
now. Yo.
Well, thank you again, everyone, for being here. Very much appreciated. So thank you all for those that attended the 20th Annual Research Symposium. The presentations this year, not only did we have a record-breaking number, but they were all really, really good. So congratulations to all the students and presenters. And now we'd like to present the Jerry Covert Student Research Awards. If you don't know who Jerry Covert is, he was the original founder and executive director of the Regional Science Consortium. So
Now they can't get rid of me.
no, now he's stuck here, whether he likes it or not. So, okay, the first place for undergraduate poster is Annabelle Aaron from Mercyhurst University for the genetic analysis of Bacillus subtilis reveals novel regulations of tolerance to high salinity. Congratulations, thank you for coming. All right, second place. Second place undergraduate oral presentation is given to Carrie Selena from the Penn State University Barron College for the evaluating the population of movement around gobies in Elk Creek, a Lake Erie tributary. All right, first place oral presentation from an undergraduate is Bryce Hansen from Mercyhurst University for the characterization of bacteria from wild mushrooms in Allegheny National Forest. Nope. All right, on to our graduate students. For poster presentations, the first place goes to Braley Motor from Mercyhurst Applied Forensic Sciences for the application of forensic archaeology to indoor crime scenes. Second place oral presentation by a graduate student goes to Ryan Zimmerman from LECOM for the comparison of UV light on colony growth on cell phones between Staphylococcus aureus and E. coli. Ryan's not there. And finally, we have our first place graduate oral presentation is Max Strile from the University of Buffalo for characterizing one of Western New York's most diverse mussel assemblages.
You know, these people are here.
It was tough. It goes on there, so
Okay, so in closing, it's been a crazy 3 days. I would like to thank all the participants of the symposium this year, the researchers, professors, especially the students. You guys did a lot of hard work getting your presentations together and doing all this research. It's awesome. I'd like to thank the DCNR staff of Trek for all the preparations, the tables, the moving things around, all special thank yous. And would like to thank, if you enjoyed your meals, it was Make It Fabulous Catering. She's awesome and always gives really good food and wonderful caterer. I'd like to thank Eric Dye over here, our silent partner in crime, for live streaming everything and making all the IT work. It's a huge relief not to have to worry about this. Just be like, Eric, okay, and then, of course, I want to especially thank Jen and Sarah and Sean and Jacob and Haley, because this is a huge team effort to put this on, and everybody, you guys worked really hard. Thank you so much. Thank you. I hope you enjoyed Symposium 2024. Come back next year, same time, same place. It'll be November 5th through 7th, and we'll be sending out the call for proposals come August. So work all summer on your project so you can come back and tell us more. Thanks, guys. Thank you so much.